clinically meaningful versus important. This included a recommendation for patient and care partner reported outcomes that could become a standard clinical trial measure. We heard about the promise and limitations of existing intervention targets, including limitations of current disease-modifying therapies and improving cognition, what matters most to most participants. We also discussed excitement around new blood-based biomarkers like amyloid and tau, but also the need for better biomarkers that align with changes in what matters most to participants, limitations of cut points and enrollment criteria in diverse populations, and opportunities to address measurement needs in diverse populations. We heard about the complexities of managing our medical system to get and provide care for everyone, but especially for aging individuals with limited access, resources, and education. And we discussed the development of measures and models that quantify the short and long-term meaningful health and financial benefits of treatments, which could be used by various stakeholders. Today, we'll hear from our lived experience expert panel with an emphasis on the question, what does this mean for me? Before we engage in our breakout sessions, where we'll have a chance to discuss measurement gaps and opportunities for what matters most: cognition, everyday function, biomarkers, and health economic, payer, and regulator considerations. Finally, we'll come together to do the challenging work of integrating the various perspectives shared to have a more representative measurement approach that captures meaningful change for person-centered AD and ADRD trials, including those focused on prevention, non-pharmacological, pharmacological, combined therapies, disease-modifying therapies, and care. As a reminder, please use chat for questions. You'll have an opportunity to join a breakout session of your choice. We'll break at 12:35 p.m. Eastern Time before hearing breakout session reports led by our moderators. We'll have a final general discussion and we'll close at 2:15 p.m. Eastern Time. Up first, Ms. Sarah Walter and our lived experience expert panel. Sarah?
Well, good morning, everyone, and thank you so much for having us here. It's really my pleasure to facilitate this panel of experts in lived experience. I'd like to start just by thanking and recognizing the members of our ACTC Research Participant Advisory Board. Can we go to the next slide, please? My co-chair, next slide. My co-chair, Lupe Morales, is here today and has been very active in the chat. Good morning, Lupe. The group that we formed, next slide, please, was formed in 2020, and we've really evolved a deep and collaborative partnership. Thanks so much to them for their generosity in sharing with us researchers what is important to them. Our group is small but mighty. It's formed of people both with research experience and people that are research curious. We have people that have been in clinical trials and those that are in observational studies. 2 of our members have a diagnosis with memory impairment. 2 have some memory concerns with no diagnosis. We have 2 that have been involved in preclinical Alzheimer's disease intervention studies, and most of them have experience providing care or support to somebody with a diagnosis of dementia. Thank you for being with us this week. It has been an incredible workshop, and your voices have been critical to help us move towards clinical meaningfulness in research. So the question of whether we have measured clinical meaningfulness or what is important to me is coming up now as people with dementia and their families consider treatment options. And when we ask people to consider their treatment options, the questions they ask reveal how they take these group level measurements of clinical meaningfulness from our research studies and apply it to themselves as individuals and families in the lives that they're living now. Next slide, please. So recently we asked our advisory board to imagine that they had a choice between taking two drugs. Both drugs are expected to help you with memory and thinking. The second drug also slows brain changes down by a small amount. Would that slowing of disease be important to you? We took their responses, which were sent by email. Some were very brief and some were long. We evaluated them for the themes and are sharing some of that preliminary data here on the slide. I think the first and most important point that was clear from their responses is that this is a complicated decision. And I'm just going to read one of the emails I received back. This is not a simple question to answer. The ability to slow disease progression would be important if all things are equal, but they seldom are. Other factors, such as the risk of taking the drug, how it interacts with or affects other medications I'm taking, the course of treatment, the cost, insurance coverage, how strong is the evidence that it works for people like me, who is recommending it, are all part of the equation. So the themes you can see that came out of their responses include motivators, accessibility and affordability, safety, the evidence needed, so what are they looking for in future research, influencing factors involved in their decision, and alternatives. So with motivators, we see some expected results here, of course, that patients and families are looking to delay the loss of brain function. They want increased quality of life with longer life. In fact, two of our participants, both of whom have the diagnosis, emphasized that extending time without quality of life would not be a motivator. One participant who had some concerns about his memory but no diagnosis said that even if he didn't see evidence of change, that he would still be motivated to take a drug that was helping his brain. Accessibility and affordability of these theoretical drugs was raised by most of our participants. This includes the cost of drug, the insurance coverage, the cost to monitor safety, how that drug is delivered, whether infusion, injection, or pill, the impact taking the drug has on others in their lives. So do I need to bring someone with me to my appointments? How frequently they are monitored, and other costs like time off work and travel. Safety concerns were, of course, raised, including wanting to understand how the drug works and how that affects the brain, whether side effects were known for people like me, including those with 2 copies of E4 allele, your ethnic or racial background. One participant said he preferred not to take a drug that causes changes in the brain due to safety concerns, and interactions were raised by 2 participants. So multiple factors influence our patients and families when they decide between drugs. And what was commonly raised was who is recommending the drug, their trust in that physician, the physician's experience with the drug, how commonly the drug is prescribed, and whether multiple options are being offered. One participant mentioned she would want to learn from other patients and families directly what their experience with the drug was before taking it. Participants also raised alternative therapies. 2 mentioned they would prefer a drug that stops Alzheimer's in its track or a cure. They also mentioned the importance of lifestyle, diet, exercise, and faith in combination with drugs. One other quote I'd just like to share from a participant: stopping dementia in its tracks or totally dissolving the plaque would of course be my choice. Slowing it to me means it's just going to take longer to fully develop and get worse as time goes on. The final theme that emerged from this brief survey is where our advisory board members were looking for further evidence. And I think this is important to us as we design clinical trials now or in the near future. How well these drugs are working in specific populations. So people want to understand, do these drugs work for people from my background? How safe are they in those populations? Do we have head-to-head comparisons between multiple drugs? and then understanding how the drugs work in combination with lifestyle modification, and specifically, how much time will be gained. It doesn't make sense in my mind to take a drug at a sizable cost of not just money, but also the time, and all along with the known and possible side effects in order to give me a few additional days or weeks. They're looking for months and years of quality life. So thank you for allowing me to share that preliminary data. I hope it builds on the data that was presented by others in previous days. And now I'm very excited to turn to our lived experience panel. We have 3 individuals joining us today: Barbara Busted, Rochelle Long, and Cynthia Hewling-Hummel. So we'll start just by asking each of you to introduce yourselves and what has been your lived experience with dementia and Alzheimer's disease. Can we go to the next slide, please? So Ms. Barbara, welcome. Can you talk to us a little bit about your experience with Alzheimer's disease and dementia?
Thank you very much, Sarah. So happy to be here again. I have been enjoying this conference very much. My name is Barbara Busted, and again, greetings from Madison, Wisconsin. My sense of all of this today and the last couple of days has been to gain that understanding. I have not had a diagnosis of Alzheimer's, but I have been involved because part of my job as a daily money manager was working with older adults and veterans. And one of the things that I found is that with one of my clients, one of my first clients, he had Alzheimer's, and that wasn't diagnosed right away, but I began getting into or starting the journey of learning more about that. So I am myself an African-American woman, 72, and I'm very happy to say I'm a clinical social worker, retired, have a blended family, and I have just a lot of joy in my life, and I really appreciate that. But I see around me so many who have been impacted by this disease, and as a social worker, even though I'm retired, I wanted to be involved. I'm involved with the UW Wisconsin Alzheimer's and Dementia Research Center. I realized it hadn't been 7 years, it's been 10 years. And I'm excited to be involved with the Get Moving project, the AFAM, which is African Americans in Midlife, the prevention, of Alzheimer's, and also the Black Leaders for Brain Health. So part of it for me is just being able to do my part, because the person in the top left, Kathy, was someone who was a great friend of ours, and she passed away from early onset Alzheimer's. And that's when I really dug in. I've been working with people, I've been doing workshops and talking about this, but it wasn't until it hit personally to me with the friends that I could actually see the devastating, effects of what this disease can do. So that's me for now, and I want to pass it on to my panel, my other panel participants.
Thank you. And we have Rochelle Long joining us from Cleveland. Can we go to the next slide, please? So, Ms. Rochelle, can you share a little bit about your experience with Alzheimer's disease and dementia?
Good morning. My name is Rochelle Long. I am the daughter of Alan and Shirley. My father was African American, my mother was white. I not only inherited my good looks from my parents, but I have ApoE gene, ApoE4 from each parent. We have 2 generations so far. I've lost 4 aunts and uncle. My father had some issues, but a heart attack took him out early. My mother has been diagnosed for 17 years. I've been living with, as a caregiver with Alzheimer's for the last 20 years. I had her sister, and for 8 of those 13 years that her sister was alive, both my mother and my aunt were diagnosed. At the diagnosis of my aunt, one of the doctors asked if we would be interested in clinical trials. At that point, it was a no-brainer because this was on that side of the family. At that time, I did not know that it was on my father's side. And so it is the only way that I can help fight. I can't do anything against this disease, but avail myself to clinical trials. My mother and my aunt both were in 3 actually, and I was their study partner. And I myself have been in the generational GTD 025, and I've been in ADNI 3 for several years. I can't remember when I signed up, but I've been a part of ACTC for a couple of years or so now, and I am part of the Minority Advisory Board for Cleveland Alzheimer's Disease Research Center. I live and reside in Shaker Heights, Ohio. I have vowed to whoever will listen to share the importance of becoming a part of the solution, not just from the complaining side. Realizing that, and I never hide from the history that the African American community seems to focus on more than not, and that is the way they've been treated. And I say they, up until about a year ago, I had never experienced a slight from the medical community because of my color. As you can see, I probably don't look like the rest of my siblings do, but I was treated very differently. And I had a problem with it. I did share that, and that won't happen again. I promise them. But I'm here for as long as I can be. I'm kind of like Barbara. I have to write things down now. I'm not worried about it, but I do think about it because it is a part of my everyday life. Every day I get up at 5, I get my mother together, I get her and drop her off at her adult day program. I come to a full-time job for the city of Cleveland. I work for the legislative branch. And then when I get home, I'm back on duty for my mom. So I live with it every day, doing the best I can and encouraging others to do so also. Thank you.
Thank you, Rochelle. Thank you. Can we go to the next slide? Welcome, Cynthia Hewling-Hummel. Thank you so much for being here. Can you talk about your experience with dementia and Alzheimer's?
Yes, well, good morning, everyone. And for those who haven't met me, I am the Reverend Dr. Cynthia Hewling-Hummel from Owego, New York. I'm a retired Presbyterian pastor. I'm a singer in a rock and roll country band. I'm an author and an artist. I'm a grandmother and a dementia advocate. I'm currently serving as a member of the National Advisory Council on Aging, NACA, and on the Alzheimer's Clinical Trials Consortium. And I'm also on a dementia advisory board for the World Health Organization. Closer to home, I regularly speak to service organizations about living well with a dementia diagnosis. I am passionate about Alzheimer's research. I enrolled in a study called ADNI, you know it, way back in 2010. I'm waiting for ADNI 4 to start up in the near future near my site in Rochester, New York. I started having memory problems back in 2003 when I was 49 years old. And I went from doctor to doctor searching for answers. The specialists I saw back then didn't have the tools we have now. And they thought the symptoms that I had might be related to menopause, a head injury, thyroid issues, radiation as a child. They suggested backing off some of my activities. I knew something was wrong with my brain, and I just kept searching for answers, going from doctor to doctor. It took me 8 years before I was given a diagnosis of amnestic mild cognitive impairment due to Alzheimer's disease. It wasn't a surprise, but it was a sadness. My mother and her only brother died with Alzheimer's disease, and my maternal grandmother has some sort of dementia. I had to leave full-time ministry because I couldn't remember my parishioners or what they'd shared with me. I loved being a pastor, and that news was devastating. But one of the ways that I was able to move forward and to redefine my life was to enroll in an Alzheimer's research study. I called the Alzheimer's Association 800 number trial match, and I was matched by Katie with a study in Rochester, New York called ADNI. Participating in research was one way that I could fight my diagnosis. Those of us who participate in research, clinical and observational studies, we undergo various procedures, MRIs and PET scans. We fast before blood work. We undergo lumbar punctures. Being in research can be physically, psychologically, and emotionally draining. And yet thousands of us have stepped up to help find answers. We participants have different motivations and reasons for enrolling in dementia studies. I myself hope to learn more about my cognitive health and how much time I have left before I'm no longer able to care for myself. My hope is that what is learned from ADNI and other studies will help protect my children and my grandchildren from developing Alzheimer's or related dementia. Thanks, Sarah.
Thank you, Cynthia. So can we stop sharing the slides now? And then I'll just turn to Barbara and each of you just to ask you to share a little bit about your research experience, what studies you've been in, and just highlights of what's been good and maybe not so great about the research you've been a part of. Barbara?
Yes, well, thank you so much. And I can relate to some of the things that both Cynthia and Rochelle have said, because even though I haven't had a diagnosis of Alzheimer's, some of those concerns, some of those fears, all of that, is applicable. What I'd like to talk about really is that I'm part of that Wisconsin Alzheimer's Prevention, the WRAP program, and a clinical core program I've been involved since 2014. And at 2015, I did something that, as far as I'm concerned, was the scariest thing ever when I made a decision to donate my brain, because I felt like, again, I needed to show others, particularly folks in my community, in the Black community, that it is possible to do something even when you think you don't have anything to give. So my experience has been, I've been welcomed, I've been encouraged by the team here at UW, Wisconsin, and I have been involved with recently the Black Leaders for Brain Health. And that's a group of Black leaders headed by Dr. Alex G. here in Madison, Wisconsin, and incredible Dr. Fabu Carter. The outreach specialist to the African-American community has been wonderful. I'm part of that, lumbar puncture and MRIs and all of that. And I talk about that with our group. We have a Get Moving group. So it's not as scary when you know you're part of a community. And we come together for all kinds of events, and we talk about our experiences, so we're not isolated. But we also talk about the fears that we have as Black folks who, we question things. So even when the researchers that we have developed trusting relationships with, when something goes amiss, something goes wrong, we bring it up because we want to be authentic, we want to be fully transparent, and we want the same from them. And sometimes it isn't easy because they're providing us with this wonderful, research and all of that. And how come we aren't signing up in droves like folks, in mainstream? Because of the fear, because of the access, because of what are you going to do to me? Dr. Bryant talked about when he has people come in to do their PET scans, a loved one is with them. They can watch outside. That's huge. I couldn't even believe that. So anyway, my experience has been very positive. I get results afterwards, but I didn't know what to ask. Now, after this conference, I'll be asking more specifically about the tests that I've undergone. The folks have been supportive, but I'm thinking sometimes because I do function fairly well, it gets sort of, get you through really quickly. Now I want to dig deeper, and my memory issues and concerns, I want them addressed. So thanks to this group and thanks to this conference and in Boston, I'm able to have more questions than just going through, yes, I'm doing okay. Thank you.
Thanks, Barbara. Rochelle, what's your research experience been like?
Well, from the very beginning, I worked with a group in Beechwood well, they were actually in Cleveland before they moved to Beechwood, but I spent many years, probably most of my experience with clinical trials, with the same group of clinicians, nurse, Dr. Alan Lerner, up until he retired here recently. And I felt like I was a part of a family. We talked about children, grandchildren, recipes. We had cake and ice cream to celebrate. I was awarded the Cornerstone Citizen Science Award by GAP in 2019. And so when we got back, they got a plaque and I got a plaque. And so we had cake and ice cream, and we would often talk about, what's happening, what's new. And it was a very good experience until I signed up with ADNI and COVID. COVID and ADNI separated us. And so we talk over the phone, but it's not the same. I had to go in for one of my MRIs and scans, and it was very sterile, detached. The clinician or technician was not having a good day, and I just felt like I was being passed through and on to the next. And that wasn't a good feeling for me, because I'm a people person. And I don't like I just didn't feel like I had any value other than what they wanted from me. And that, if it hadn't been for the group back in the office, I might have been done, because there's nothing there for me, then I don't have anything to give to you. My passion for this disease helped me to get past that. Knowing what my future is, the chances for me went up. And so I've got to do what I can do, not just for myself, but I want to make sure that my great-granddaughter doesn't even have to think about it. Thanks, Sarah.
Thanks, Rochelle. Cynthia, can you share with us a little bit about your research experience?
Of course. So my research experience has been mixed. I love my study team, but at the same time, ADNI is a very lonely study. In all my visits over all the years, I never saw or talked with any other ADNI research participants. It's hard not to be jealous when I hear of Barbara and research studies that are having luncheons and meet and greets where participants connect and they share with researchers and one another. When I enrolled in ADNI back in 2010, I asked about the possibility of meeting other study participants, and I was told it was not possible because of privacy protocols that were in place. It wasn't until I joined this group, ACTC, about 4 years ago, that I actually met somebody else in an ADNI study, my friend and fellow advocate, Rochelle, who just spoke with us. I heard through the ACTC connections that many study sites were sending out regular newsletters to study participants, highlighting research updates that came out of ADNI data. And I wondered why I never received a newsletter or anything from ADNI. And I was told that the local IRB, the site, would not allow it. Well, something must have changed somewhere, because I finally got my first official ADNI newsletter in 2023, 13 years in, that showcased ADNI advances. And in my mind, regular communications to participants should be built into all studies. I know I'm further motivated when I hear something was learned and that my participation made a difference. And here's something else. When a study ends abruptly, participants should not learn about it on the local news. Many of us are motivated to participate in dementia studies because we want to know more about our own results and what they mean for us and for our families. I stumbled onto my patient portal when I had a procedure that was at the same hospital where my ADNI MRIs and PET scans were done. And suddenly I was able to access images and impressions along with comments and interpretations. And it was amazing to have access to all of it. But at the same time, I was afraid that I would be caught or get into trouble for looking at my own health information. And I really longed to have in-depth conversations about what my results meant. I've heard that about other studies, that they have regular meetings with participants to share and review health information. I realize not everyone wants to get test results, but some of us do. And it should be more than an option. It should be a right that's built into every study.
Thank you all for sharing your research experience. So now turning to the topic of this workshop, with this experience, which of the outcomes that we discussed this week are clinically relevant to you, to your family, to your community? Barbara?
Yes. I did I was trying to do a survey of our Get Moving group, but we were moving so much with our exercising, and then they had a little luncheon set up for us, so nobody wanted to just talk. It was just like, let's get to the food. But over the course of years, I have been able to have some really deep conversations with a number of folks that I've known. Either they've been part of the study and also members of my church, as well as in the community. And so the blood biomarkers, they are a game changer, because one of the things that people talk about is not wanting to go in for an MRI. Even though we can get results of that, and someone actually, they found an aneurysm and she got that treated. So she felt like that saved her life. And that wasn't what she was expecting. But the blood biomarkers are what people are feeling more comfortable with, and being able to know, wait a minute, if I get this done, and this is the information that they can then figure out, do I have this gene? Do I have amyloid plaque? And in the African American community, amyloid plaque is not what it is in other, in other groups. And we're learning that. So maybe a reduction of amyloid plaque, may not be the best thing for us. And if that means, though, that we're not a part of clinical trials, now that we've been, recruited and encouraged to be a part of that, what does that say about us? Now we're kind of pushed to the side. So those are some questions that we have concerns about. But the thing is, for me, wanting to know what is happening with my life, and when I talk with other people, it's like, how much time do I have? And for people, what is the quality of life that I will have? Those are the questions that I want answered. If I'm able to give my blood and someone can test it and review it, and the research indicates that, and I want to be able to choose that on my terms, not necessarily all the information, all the data, but things. I love the Bill of Rights because that really speaks to what I hear the other research participants say. So thank you.
Thank you, Barbara. And Rochelle, what outcomes are clinically relevant to you?
Time and information. Much like what Barbara was saying, a lot of people don't want to go in the tube. They don't want stuff attached to their head. Mm-hmm. They want to know who has access to my blood. If they do the blood biomarkers, what are they going to do with all of this information? How will I know where it is or what's going on? These are things that, they feel is taken away, but what are you giving me back in exchange? How will I be treated? What happens if I do get this disease? Who's going to help me? I don't have any insurance, or the insurance I have is not accepted. And so when we talk about the outcomes in our community, there is a huge mistrust of the medical community and research. Simply, Cleveland Clinic is here in Cleveland. And I happen to they have put up a site for clinical trials by Cleveland Clinic. And they asked for some blood. And I went in to donate because that's part of what I do. But I got asked the question, how do we get people to participate in this community? Cleveland Clinic is in the heart of the city and in the urban part of our city. And my response is, you got to give them something. You just can't say, hey, give me. This is, we need this from you. When you have come and you have taken their land, you have taken everything and you've shown them big shiny things that they don't have access to. You have to establish a relationship. In order for somebody to trust, there has to be a relationship. I can tell one of my coworkers, I discovered a great restaurant, and she or he will go because of our relationship and our connected love for food. Well, when the medical community comes in and they're the big giant and they're just asking for your blood, they're not coming with any, they don't even know you. And so it's so important for a person to feel like they are valued beyond what the research community can get from them. Where are you going to be if I get diagnosed? Who's going to be there for me? And, you know, as I try to explain that best I can. But we still have a ways to go.
Thank you, Rochelle. Cynthia?
I would say it's all relevant to me. I want to know what's changed and what the changes mean, especially in regard to my cognitive health. What score went up? What score went down? And what does that mean to me? Thanks to 23andMe, not ADNI, but 23andMe, I know I'm homozygous A4. And it's good for me to have that information because there are new treatments that are not appropriate for me at this time. There are new biomarkers that are being discovered all the time. And I want to know what's changed since my last test, since my last scan. I want to be able to track and discuss my results of, especially of my cognitive testing. And I know it's harder and harder for me to remember. And I want to understand why. What are the physical reasons that are causing the changes? And what can I do to slow down the progress of this disease? My theory is this. If researchers are tracking something about my brain or in my cognition, whatever it is they're tracking, I want to understand why, because that's clinically relevant to me. I just have a need to know, where do I stand?
Thank you, Cynthia. So we are going a little over time, and we did have one last question for you all. So I don't know if we can make it as brief as possible, but so that we can get to the discussion groups. What would you like to see change? Barbara, you've talked about this a little bit, but what would you like to see different in research?
You know, just that the researchers implement culturally sensitive and relevant practices in working with research participants and utilize the current research that shows the importance of going to the community. Like I love Dr. Sims, you know, going to the community rather than expecting more marginalized individuals to go to clinics to participate in research. That's the biggest one for me, those 2 points. Thank you.
Thank you. And Rochelle?
Dr. Sid O'Brien yesterday said it perfectly. To take it into, and Barbara just take it into the community so that they can see that here's a part. We want to be a part. We want to have that relationship. I would like researchers to develop a relationship long-term, not just for the time frame of getting what they need. To treated like I matter. And I know I keep saying that, but value is important to us, even after we're diagnosed.
Thank you, Rochelle. And Cynthia, I know you'd like to share the proposed Bill of Rights for sharing research results, so if we could pull up those slides and go ahead.
Before we get to the Bill of Rights, I'd just like researchers to treat us as partners in a study rather than subjects in an experiment. I would like researchers to open opportunities, more opportunities for those of us who live alone, who want to participate in research, and to use technology to make it easier for study participants and partners to check in between visits rather than trek into a facility. Bring the studies to the communities. And I would ask researchers to build relationships with study participants, to ask our thoughts, get our opinions on priorities. And if we don't qualify for your study, please refer us to another study or maybe even a virtual online study that we could make a contribution to. But most of all, I would like them to pledge to adopt the study participants' Bill of Rights. And I want to share those with you right now. I have the right to receive my individual results collected in the course of my research participation, if I so choose. I can also ask how to receive them. This may be done in person or by telehealth, either alone or with a person of my choosing. I have the right to exercise this right in an informed manner, including receiving information on validated decision-making tools if they are available, knowing who can access my results, how the law does or does not protect me after receiving my results. In order to protect myself, I may need to finish any care, legal, and financial planning in order to advance the receiving of my results. I have the right to be told what my test results mean in an easy to understand term with sensitivity, compassion, and patience. This information should also be provided in writing so that I might review it later. I have the right for my questions to be answered to the best of the researcher's knowledge and to take all the time I need to process the information I received. I have the right to be contacted or decline to be contacted to check on my well-being after receiving a result suggesting an increased risk of dementia, and to be referred to additional resources for more information and support related to my health and well-being. I have the right to decide what actions I take after receiving my test results, such as pursuing medical and/or psychological care, engaging in legal or financial planning, and informing my family and my friends of my results. I have the right to turn my results into action for my own well-being and the betterment of others by exploring additional research opportunities that I may qualify for. And the above rights should apply regardless of my cognitive status. Thank you very much.
Thank you, Cynthia, for working on that Bill of Rights with us as part of the agreed, and I'm happy to share the link in the chat. Apologies to those of you for going over, but this has been an incredible panel. Thank you, each of you, for taking the time to share your experience and to your honest feedback. We can only get better if we are talking to you and listening to you and partnering with you. So thank you.
Thank you.
I'll turn it back over to Luke.
Yes, thank you, Sarah, Barbara, Cynthia, and Rochelle. That was a very powerful session, and we appreciate your time and perspectives. And to everyone who has also shared in chat, we appreciate that. And if the panel hasn't had a chance to review that chat, I'd encourage you to do so. We're going to switch now to our breakout sessions.
I'm trying to make this be a switch emotionally to, talk about cognition at this point. That was an amazing session. Really good. Thank you so much. And we'll give everyone just another minute or so. Christy, it's so nice to see you, Melissa, Christy. And hi. Great. I believe we have about 24 participants joining. and we'll be here for about another 58 minutes or so. And I'm sure there's going to be so much to talk about, a lot of meaningful conversations.
Am I
And
supposed to be here, Rosie?
I'd love you to be here. Because you were part of the conversation or a speaker in the cognition session, it would be lovely if you were here. But if you're really interested in another session, I don't know. I don't know what the rules are. Christy, do you know?
No, I'm happy to be here.
Yes, there are no rules. We would love to have you here, but please don't feel free that if you want to explore the other rooms as well, you can, but it's great to have your.
I'll
Great.
stick around.
Terrific. So I think we can start introducing ourselves. I am one of the moderators of this session. My name's first on the screen, so I'll jump in first, but I'm not necessarily the lead moderator. In fact, one of the goals is to really hear as many voices as possible. My name is Dr. Rosie Curiel-Sid. I'm an associate professor at the University of Miami, and I'm an Alzheimer's disease researcher and clinician. And I develop new cognitive measures and for the early detection of Alzheimer's, and using the best that cognitive neuroscience has given us. And I try to relate those cognitive changes and cognitive transitions to biomarkers of disease. And so it's just a pleasure for me to have joined this group for the last 3 days. Thank you so much. And I'll pass it on to Dr. Christy Hardy.
Hi, everyone. I am Christy Hardy. I am a program director in the National Institute of Neurological Disorders and Stroke, and I'm also a practicing neuropsychologist. I apologize for my background. I'm in a hotel in Chicago, which is not where I have been for the rest of the meeting, and I didn't have time to change my background. But I am really excited to have this discussion with everyone. And as Dr. Sid said, we really, really would love to have everybody's participation in discussing our topics for the next hour. Melissa?
Hi, everyone. I'm Melissa Trevino. I'm a program director at the National Institute on Aging. By training, I am a cognitive psychologist. So thank you, everyone, for coming to our breakout session and getting started. So
Great.
I'll turn it over to Rosie.
Thanks. So for the purposes of time, we won't go through introductions for everybody in the group because but we do encourage you to please introduce yourself in the chat so that as we can speak, everyone has an opportunity to share your your role as a stakeholder. So please feel free to introduce yourselves in the chat. So the primary goals of this, and we'll talk about what's going to happen. The primary goals of this session is, as Christy said, to hear from as many people as possible, particularly if you still haven't had a chance to voice your input, or if you have addressed something in the chat, but it still hasn't been answered, or we need to unpack it a little bit more. Please feel free to do that. We also want to someone's going to put up on the screen the gaps and opportunities slide that we started off with. There we go. Thank you so much. That we started off with. We're going to go through these gaps and opportunities briefly again, and then we're going to modify them. Maybe we're going to make them a little bit more specific. Maybe we could, improve upon them together. So that would be a wonderful goal. I'm going to be taking a lot of notes. I'm not going to be, well, I can't promise I won't be that active because I tend to run my mouth, but I want to hear from everyone else. But I will be taking a ton of notes, and hopefully we can modify this and make it even better together. And then after this session is over, the moderators will try to take this back to the main group and share any takeaways in about 10 minutes or so. And we'll have a general discussion at the end of the breakout session. So that's what the intention is. So unless there's anything my co-moderators want to add to that, we can jump right in to talking about cognition and some of the gaps and opportunities that were generated before this meeting began. And particularly that's really related to the work of some of the speakers in the cognition session that happened on day one. And we're so lucky and fortunate to have some of our speakers here with us. I see Dr. Gibbons. Oh, all of them are here. Dr. Martinez, Dr. Jarmin, thank you so much again for connecting and being here with us, because we're going to be able to listen to some of our other citizen scientists, as well as some of our other clinical researchers and clinical scientists that we can help refine this together. So the first was Dr. Martinez's talk. And I was wondering, Dr. Martinez, if you wanted to read the first gap and opportunity, which is equity in cognitive measurement.
Yes, happy to. Thank you for doing this session. It's been such a fantastic workshop. I've learned so much throughout of it. So thank you for everybody who put it together and my co-presenters. So yeah, so the gap number one, I'll just read off from the screen, equity in cognitive measurement. So there's a need for skillful and informed adaptation and cross-cultural validation of cognitive tests that measure clinically salient outcomes. There is a lack of harmonized longitudinal assessments of diverse Americans. So that's what we're seeing as an important gap, and some of the opportunities related to that, and some becoming a little bit more specific, is that this includes developing novel cognitive testing approaches and technologies that are sensitive and specific enough to detect and or track clinically meaningful change over time that is indicative of disease progression. And then to facilitate collaborative research amongst investigators that study how AD and ADRD presents and progresses in diverse older adults to optimize and accelerate the availability of valid and reliable cognitive measurement.
Great. And your talk was mostly about this, Dr. Martinez. And I wanted to kind of start, perhaps you would start with leading some of what some of what we read here, and some of the things that I remember from your talk is, making sure that we have cognitive measures that are adapted or developed from the outset with persons from diverse groups, but how hard it is to do that sometimes unless we work together, because we don't have access. Some researchers just don't have access to these populations. I'm very fortunate. I work at the University of Miami. So we have people from all over the world. And that helps. That's a wonderful platform for me to do my research, but some researchers don't have access. So perhaps you want to talk about that a little, and others can comment.
Yeah, I think you hit one of the main points that I was trying to make during the talk, and I think were made by other investigators in other sessions as well. I think every single session, actually, this point was made, is the importance of representation in research, and that persons from all backgrounds are represented in the work that we do, both in the clinical trials, in the development, at every point of the research enterprise. That I think it's important that everybody is included, that voices are being heard. And I think obviously this is not a novel concept. We thought about it, we know about it for a long time. But I think only recently it's become extremely obvious to a lot of people, although not all, how important that is to produce the excellent and best science that we possibly can, that can benefit everyone. So I think that's something that sometimes we don't always keep in mind, is that we really need to have that diverse representation to do our best science. And most importantly, because that's just science, that's just what's right, that everybody should be represented in the research. And so I think that's a point that I wanted to make. I think other things that we didn't quite get to that go beyond that is making sure that we're developing the infrastructure and the support systems to have that happen. And I always think about this in my own work. I developed my research career at the University of California, San Diego, in Southern California, where there was a large Latino representation. And I recently joined here Duke, which is in North Carolina, with a much Neuro-Latino population. That's a population I focused on for various reasons. But obviously, I think all populations should be represented. And I think it's extremely important that we think about the changing demographics in the US and the systems that need to be put in place to make sure that this research can happen everywhere. So just as an example, there's a large rural population here and how to access that population and how to serve that population. I think like we talk about kind of how can we get people in, and I always try to think like, what can we do for the population first? Like we shouldn't be asking something, but offering something first. And that's how I try to start all of my interactions, just because I think it's the right thing to do. So I'm going in circles. I'm not sure how helpful this is for the group, but I just think that the core things to make sure that come across is that representation is crucial at every single level. And we need to have diverse representation across the board. So that's the main point I wanted to make.
Dr. Bakini, I have a follow-up question. A big thing that I've been thinking about in terms of equity and developing these measures is how we can be more inclusive of people who are speakers of other languages than English. And, I think a lot of our measures are not translated at all, or not translated in a way where we can be certain that the translation is valid and reliable across different groups. And what do you think and it's expensive and time consuming to do these translations. So do you think that as we move ahead in this space that we should just be really trying to devote the resources and time that we need to develop some of these really gold standard translations for many different groups here in the US, but also other places where we need to be reaching people who don't speak English because they have been largely left out of our studies? Or do you think we should really be working more to include those speakers of other languages in studies that involve totally different technologies that get at cognition in different ways, like wearables and device using, things like that?
Yeah, I think the short answer, I think we can and we should do both. I think it's feasible. I really think it is. I think that a lot of the same principles that applied for kind of traditional cognitive tests apply for how we're going to interpret results from novel technology. It's another way to capture cognition through the behavioral manifestations. And I think we should do both. I really think, a lot of my newer studies have to do with technology, and I see tremendous promise in that. I think we also should not forget some of the standard ways that already are implemented and in use in majority non-Latino whites population, and that we just don't even have that in speakers of other languages. We don't have the basics. And I think they are still extremely important. So for example, when I presented that information on how we adapted to Spanish and created new normative data and so forth, and some people see that and say, well, see, the tool is not helpful. We've been doing it wrong. And I think what we need to remember is that it's about how you use the tool. So it's like if you have a knife, right, and you're trying to cut with the non-sharp edge, it's not going to work. It's not going to do its job. So some of the tools that we've developed before, there are aspects that work. There are aspects, there are principles that were developed that are tremendously important that as we move forward. I think we should move forward. I think we shouldn't be stuck in what's, what was before. I think we should build from that and add these newer ways to make sure we produce the best science that we can and the best tools that we can. So I know I think it really can be both, and it can be both within the same study. I think it's important that it's both within the same study, so that we can advance both parts of it. I always wonder with the technology that I get, again, I'm all for, I really am. I think it's tremendous. It's a tremendous tool, is that we need to be testing, okay, how much more helpful is this compared to our standard thing? Is this more helpful or less? And I don't think we have an answer to that. I think we need to find an answer. I think it's an empirical question.
But we have found answers to some of it. So we do know, for example, And let's debate about this if possible. We do know that some measures are more superior than the standard in detecting very early cognitive changes. The NIA has invested millions and millions and millions of dollars in research to develop new early detection tools. And these have not been adopted by the scientific community. We're still using paradigms that we did during the Army Alpha era. And we could do better than that. So I believe that I agree with you 100% that some basic measures have to be translated so that immediately we have as much basic as possible. But when it comes to moving forward with novel measures, I'm cautious about dedicating a lot of resources to mega translation efforts of measures that are just not that useful. In particular, they're good for detecting dementia, but they're not that useful for detecting MCI or earlier states, and they have minimal to no correlation with biomarkers. And so, I'll be quiet so that Ms. Cynthia can speak, but that's sort of my counterpoint to what you're mentioning.
I just wanted to say something about cognitive testing. To me, that's the hardest and the worst part of being in a study. I would rather have a lumbar puncture. I would rather do anything else than have cognitive testing because I feel so stupid. Here I am with my doctorate in ministry and I can't answer basic questions. It's heartbreaking for me to go through that. I often sit through the testing in tears. But when you talk about connecting it to biomarkers or the changes in the brain, then I'm not beating myself up because it's something that's physically happening in my brain that I don't have necessarily have control over if it's a disease or something that's changing. So I think that the cognitive testing to just be aware of the difficulty that it presents for many people who want to volunteer to participate in trials. And when I think about cognition and how you measure it, to remember that it's very difficult for many of us who are sitting in those chairs waiting, to kind of just get through it.
Yes, thank you for sharing that. It's a lot of pressure sometimes people feel. Thank you, Dr. Germain.
So thank you for making that point, Dr. Hummel. I think what's interesting is that doesn't just reflect a hardship of being in a research study that's necessary and unavoidable, but in fact reflects that many of the tests that we're delivering in these trials are not well calibrated to the current abilities of the person taking the test, right? Like in a perfect world, we should not have cognitive testing that feels so incredibly difficult to you as a participant that you come out feeling like you couldn't accomplish anything, right? We should be able to calibrate difficulty to get at where you are at the time of testing. And, Dr. Gibbons made this point in his talk as well about adaptive testing. But I think this is one of the big challenges we have as a field is we're using a one-size-fits-all approach to cognitive testing that isn't appropriate for a lot of patients. So,
So I also wanted to follow up with Cynthia. Cynthia, thank you for sharing. I had a question for you, which is, we've talked in other sessions, and you guys have talked about this as well, but someone's got to mute. So we've talked a little bit about remote cognitive testing at home, where you're not going to necessarily have someone sitting across from you. And I'd be interested in your experience sitting across with another person doing the cognitive testing versus doing some cognitive testing on your own in your own home. Is there a different experience you have? Is there a preference that you have one way or the other?
I could say it really depends on the tester. Some of the testers, I just seem to send out a vibration of compassion. It's easier just to be at home. There's one test I think about on the computer that it gives you a sound whenever you make a wrong answer, but I can turn down the volume so I don't have to listen to that. I always just feel humiliated by what I'm not able to do that I once could do. And so it's about beating myself up.
And you'd mentioned, sorry, I just want to follow up one thing. You'd mentioned in the Bill of Rights, you want follow-up, right?
Yeah.
So you talk about beating yourself up, and what I hear is that's, coming from you. It's about your own performance and your own expectations for your performance. I mean, the beeping sounds are probably not the most helpful thing in the world as well, but what would that follow-up look like after taking that testing for you?
The follow-up would look like, here is a graph of all the different tests we've given you over the years. And this is where we are today. And you can see what's gone up and what's gone down. And this is related to this change that we observed in the MRI or the PET scan. So this is where you seem to be, so that they can answer my question about how much longer can I live on my own? How much longer am I able to do these things that matter to me? So it's about planning my life out, and having that information helps me to prepare my family for the changes that are coming. My mom's Alzheimer's started getting bad in her late 70s. I just turned 70. So, I have 2 copies of ApoE4, and I'm just mindful that the time is ticking. And I want to do what I can with the time I still have.
And when you've had your results shared to date, if you've had your results shared to date, what does that experience look like to you? So not like what you're painting the picture of as your ideal, but what has it actually been like for you?
Well, I can remember sitting down with a principal investigator, going through it very quickly, but I didn't have anything that I could take home. I'm mindful, there's a lot going on in the study center, and to take a researcher's time that's very valuable takes it away from somebody else. But maybe there's somebody else that could do that with me. I can remember one time I got a call at home. It was dinner time, and I was just scrambling for a piece of paper, trying to write down information. But because my memory is so poor, I just really need to be able to have a piece of paper in my hand, and then I can go back to it and think about it. And so as much as possible, those things would be beneficial to me, and I'm sure perhaps to others.
You raise such really important points, and I think the science is there to support you, but the measurement science has not been. The ability to use adaptive testing as an example, to be able to tailor items to your particular level of ability, so you're not getting tasks that are ridiculously complicated. My illustration of giving calculus items to a child, is essentially the paradigm that exists in cognitive testing today. So being able to take your last measurement to start adaptive testing of your next measurement, so that we give you items that are appropriate to your level of ability and potentially impairment, I think is critically important. The other thing is that being able to assess people at more frequent intervals using adaptive testing, or using some kind of alternative wearable types of technologies, will also give us the kind of higher dimensional longitudinal data that will make the prediction of the future for you much more attainable. Having to go through multiple hours with a trained neuropsychologist administering the same batteries over and over again is really going to limit the dimensionality or frequency of longitudinal assessments, and that's going to limit the ability to predict into the future. So, your comments are just spot on. Thank you.
I think the other thing related to that is, even within the battery of testing on a given day, some of the questions are in multiple tests. What day is it? I just answered that a few minutes ago in another test. Or even having going to different studies, asking the same battery of tests. I know the story about the woman that loses her purse and her kids are hungry and the police help her out. There should be ways of bringing more resources to the table so that an accurate picture. I know I don't have the memory I once have. They adapted a test for me once when I went through a neuropsych, and they said, "You're a minister, name the books of the Bible." And I could only get as far as Leviticus, and I started to cry. I couldn't go further. And that was heartbreaking, but it is what it is. But knowing that there's biological changes in my brain, then I don't beat myself up.
Of course, of course.
I think Cynthia, you actually articulated over the course of, I've been writing it down, the last session and this one, 5 things that we could, I say easily, it's really low hanging fruit. So one is returning scores in longitudinal studies, what went up, right? What went down? What does it mean to the extent we know? And we don't know everything, but we know some things, otherwise we wouldn't be measuring them. What we are tracking and why in general, right? As well as your latest point, something to take home that doesn't require you to remember all of the things in that return, right? So having that in a form that you can take home and look at a time that is best for you. And, those 5 points right there, I think already give us a roadmap for what we need to be able to do for participants. So thank you for that.
I just wanted to add, my name is Lupe Morales. I'm the co-chair of ACTC Advisor Group. And I just wanted to add to that 5 is some feedback loop that you can ask participants. What was your experience like going through this process? How can we make it better? And utilize that as a continuous improvement loop. Just the human part of it, having been a research participant myself, that's the only thing I can hang it on, is that learning from the experience I had going through the process that you developed to get us through the research. It's really important. And it's just that human touch point. Thank you.
Yeah, thanks for raising that point, Lupe. I think it's tremendously important what you're sharing, and it's tremendously important that we continue to do it, because I think it obviously changes from person to person. It changes from time to time, right? And what worked one way didn't work for, next time for whatever reason that was. I think it's tremendously important. I know that we've learned a lot from our participants, especially again, as I'm transition to a new area and trying to understand the population best. I see so many differences from the population here, from what the population needs for someone else. And I think it's important that we continue to include that as a thing we do all the time and continuously evaluate it.
And really concretely, Lupe, to your point, when we have gotten feedback from participants, we had this, I have this Since Science platform that's been running for about 15 to 20 years now. We had people give stars to tests, right? Like, how many stars would you give this test or this experiment or this study? How many stars people give a cognitive test relates very well to the measurement properties of that cognitive test, right? So tests where participants are like, this was kind of awful, don't measure cognition very well. The tests where participants are like, actually, I feel like this is both engaging and it got at my cognition in a really good way, and I actually even enjoyed it a little bit, measure cognition much better. So it's not just sort of feedback so that we can be more helpful, but it's really meaningful scientific information.
This is great. Thank you so much. So we got a lot of information. I too have been trying to write things down. So I will say that this first point, equity and cognitive measurement, is now instead of having 2 opportunities, has 4, because we've this value of integrating the lived experience experts contributions to expanding upon the equity and cognitive measurement by providing follow-up results to those who want it, so that they can integrate their results into their current life and their future planning, et cetera. Learning what went up and what went down and what does that mean, what we're tracking and why, something to take home to process it in a form that is best for you, to develop a way to return results to participants, kind of like a best practices, if you will, and develop a continuous improvement loop so that we can learn about what aspects of our research can be improved by persons from different perspectives. And so all of that has really served to populate this first point of improving equity in cognitive measurement, in addition to the staple things that were discussed by Dr. Martinez about adaptation, normative data. There's a comment by Dr. Huff in the chat about developing new tests or developing normative data in more diverse samples. And I think from my what I understood was that both are very important. Really developing normative data on the tests that are working and are most useful to early detection and monitoring, but also capitalizing on the cognitive neuroscientists to develop new tests and new innovations. One of my dear friends and colleagues, Dr. David Lowenstein, he says, imagine if medicine were still using X-rays and we didn't innovate and develop CT scans and we didn't develop MRI scans and we were just using X-rays. Well, in the cognitive space, we're still using X-rays in some studies for cognitive measurement, and so we really have to innovate upon that as well. So I don't know if any other comments would like to be made by our wonderful statisticians in the group or others before moving into discussing our second point, or this could lead into our second point, which is adaptive measurement of cognitive abilities. I will say that, well, I'll give an opportunity for anyone to speak up before I say that we've already added one more opportunity to the second point through our previous discussion. That opportunity, or that point, the second point, is that adaptive testing can tailor items to give tasks that are personalized and well calibrated to the current ability of the participant. One size does not fit all, and research participants may experience negative emotions such as humiliation, noticing that they can no longer do what they were once able to do. And so thank you so much to Dr. Hummel and Dr. Jermeen and others that contributed, because that's an additional point that we're making in number 2. All right, so we can move on to number 2 unless anybody wants to make another point about equity and cognitive measurement. Speak up. This is a friendly group. All right. Dr. Gibbons, would you like to lead us, or any other statistical experts, would you like to lead us with describing number 2?
Sure, happy to do so.
Thanks.
So adaptive measurement of cognitive abilities, such an important topic. There is a need to improve upon the precision of repeated cognitive measurement by reducing practice effects, often produced by traditional fixed-length cognitive assessments, and reduce time and burden of the testing process. So in our experience with, well, I'll continue on to the bullet below it. Large cognitive item banks can be adaptively administered, tailored to a person's specific level of cognitive ability with precision, and can be repeatedly administered without practice effects, enabling high frequency longitudinal assessments across a person's lifetime. So this is really such an important paradigm. The paradigm shift is from traditional fixed length tests that sacrifice the precision of measurement for the speed of measurement to adaptive tests that enable us to fix the precision of measurement at high levels, but allow the items that are administered to vary both in type and in quantity from person to person. And the benefits are enormous. We have shown already in our preliminary work that using a small fraction of the items that are traditionally administered, and those same items administered over and over again, we can reproduce with almost perfect precision the entire item bank from a very small subset of items. Imagine being able to replace a interview, a neuropsychological testing session that might last half of a day, that requires a trained neuropsychologist to administer, with a 5 to 10 minute adaptive test that could be administered on your phone anywhere and at any frequency. Imagine the amount of valuable longitudinal data that we could have. This doesn't mean we want to get rid of neuropsychologists. Of course not. We want to direct their attention to those people who need further assessment, where our uncertainty in the estimate may be larger for one person at one point in time than another. So these are kinds of things that are that can be achieved using multidimensional item response theory and computerized adaptive testing. But there's another piece to it about equity and about some of the discussions that we've talked about in the first bullet. And that is, it would be great to recalibrate every test in every subpopulation, every minoritized population. But you can't do it. But what you can do is you can look at how the items differ in different subpopulations in terms of their performance against trillions of possible different response patterns to the items that are administered. This is called differential item functioning. And we've done this in a lot of contexts where we don't want to go out and revalidate every single measure in every single language, in every single culture, and in every single subculture. But we can administer these in those as adaptive tests and use the data from that to determine which of the items in the bank are actually biased in that population, which don't do a good job of differentiating people of high and low levels of ability. We can do that very rapidly using regular kinds of adaptive administrations. So that's another piece that I think becomes very important. Maggie Allegria from Harvard and I translated our bank that contains thousands of items for mental health and substance use disorders. And this was a huge project. We had to forward, reverse, and adjudicate the translation. But once it's been translated, it doesn't mean that it works in Spanish speakers, in different ethnic subpopulations. There we use differential item functioning to identify which items are just bad items. In depression, somatic items like fatigue are very important in a general population, but they don't tell you anything about perinatal depression. All pregnant women and postpartum women are fatigued. It has nothing to do with depression. And that can be easily identified without having to go into extensive validations. I'll stop there.
Dr. Duff?
Thank you very much. I think that there is a lot of promise in adaptive measurements, and I really appreciate Dr. Gibbons sharing his expertise with us. But I wonder if what adaptive testing is, a lot of the examples that he gave are more depression or anxiety or psychiatric symptoms, how that actually carries over into cognitive assessments like delayed memory. So, we want to collect information about constructs that we know are valuable in the determination of somebody progressing from normal to MCI to Alzheimer's disease. And so I worry that some of the, that we'll be losing information by shrinking these batteries down and, or collecting them in a very different way. For example, I think we all know as neuropsychologists that delayed recall is incredibly valuable in tracking individuals that are progressing through amnestic conditions. But delayed recall is very challenging to capture on an iPhone. Most of the tests that have been out there don't actually measure delayed recall, but they look at delayed recognition. And recognition is very different than recall. And so I worry about making some of these changes too quickly without really seeing if we're evaluating the same constructs. And just lastly, from the conversation on Tuesday, is that I also think that practice effects are not something that we should necessarily get rid of. I think that practice effects in some ways shows us a meta measure of memory. Someone's ability to learn information and then retain it after a week or a month or a year says something about their memory that I don't think we capture with traditional memory tests. So I'm a little bit of an advocate for not throwing out practice effects or reducing them, because I think it's a valuable cognitive construct. But again, thank you very much for your work. I think it's fantastic. And I do think that there's a lot of promise in many of the other areas that we collect data in Alzheimer's research. I just worry about how much it might dilute the cognitive assessment piece.
So the good thing is we now have data on this. So I can't speak to that much of the substance. It's certainly not my area of expertise, but Dave Gallo, the former chair of the Department of Psychology at the University of Chicago, is the member of our team that developed the item bank. And what we did is we did this in parallel with our colleagues, David Bennett and Bob Wilson at the Rush Alzheimer's Disease Center. And we administered the same traditional test batteries, which include delayed memory, to those patients who got the adaptive tests. And what we found is, and our initial work is on the global dimension, what we found was that we got extremely high correlation between lengthy clinician administered neuropsychological tests and not the actual items on the adaptive tests, but the results of the adaptive test administration. So we are optimistic that with careful development of these item banks, we're able to administer these tests. There are certain subpopulations like, MCI, where many of these tests are not going to be able to be administered just on a person's iPhone. They'll be administered on a computer, and there'll be the assistance of a research assistant who can help walk through the administration process. And we've learned that kind of thing in our work with children as well, in looking at psychopathology. So we're very optimistic that we can build these kinds of large item banks and retain the kind of important information that you're talking about, and hope to continue that work.
Please go ahead, Dr. Jermine. You're still on mute, and I was trying to get myself off mute.
That I would stop doing that at this point. But so having done a lot of rubber meets the road cognitive test development, including with IRT and with item banks, I will say that it is extraordinarily effective for building subtests or mini tests or whatever when you are dealing with visual reasoning, verbal reasoning, or judgment of individual stimuli where the judgment is independent of the other items. Where it struggles, I think, is with memory in which there is inter-item dependence, right? So of course, this is a lot of memory because of interference effects. Items interfere with each other. When you have a different set of items delivered together, the items now behave differently than when they were paired with other items or adjacent to other items. When we move into a high frequency longitudinal test design, that problem becomes even worse because now you have massive interference across the many high frequency assessments. And so I think this is actually like a big gap. We want to move to longitudinal high frequency assessments, but memory makes all those subtests and items behave differently the more you do it and what the items are. And it's very, very difficult. Easier in the verbal domain, the visual domain, because visual item banks are very hard to generate, especially if you want to link them to concrete knowledge. So I think that is actually a very big, big gap. The recall question, I think, is interesting because it's the easiest to do in person, right? So these many paradigms developed on recall because it's incredibly low burden and incredibly easy to do in person, very hard to do digitally. And if we want to move to a scalable digital context, those are the ones that will translate the most poorly. But those are some of our most, our workhorse sorts of tasks. So I would identify that as another gap, specifically in the cognitive domains that can be done in high frequency designs at scale.
And maybe you could just repeat that last point when we go on to our point number 3, so that we can reiterate it in that context. Thanks so much. So we are going to move into a break between 12:35 and 12:45. And so we do have a few minutes that I want to just let you know that. So we're gonna wrap this up. I'm gonna try to summarize, and you all can correct if I missed anything. There's a lot of really good stuff that came up here, and some differences. And by the way, as I'm observing everyone talking, I really just feel that these are the kinds of collaborations that need to happen, because sometimes we do work in silos. I can't possibly have the knowledge that Dr. Gibbons is bringing forth with his expertise, and Dr. Germain and I do sort of the same work, but in different ways. And so coming together, using the statistical expertise with the knowledge of the cognitive constructs and what might work and what might not work, and how we can, like you say, carefully, if we do the CAT, how we can carefully construct those item banks, making sure that we're putting measures that are meaningful in there, but also, looking at other valuable cognitive constructs. So like practice effects, which have been looked at a lot and there's been mixed data about their utility, but there's some groups that have really found a secret sauce in how they looked at diminished practice effects, and how valuable that could be in terms of early detection and association. So there was a method of looking at diminished practice effects by repeat administration in multi-day learning curves, for example, with some of our friends out of Harvard. And that's very an innovative way to look at practice effects. So I've included that as another opportunity. Is practice effects should not necessarily be looked at as invaluable, but could be a valuable construct. And even looking at practice effects in a more granular fashion there could be a secret in that sauce as supported by the data coming out of Harvard. And that there is other adaptive methods, statistical methods, such as differential item functioning, which is a technique that can cross language barriers and cultures and even subcultures, and can tell us which items in a large bank of items might be biased and might not necessarily differentiate people with high versus low abilities. And there were also mention of cognitive assessments specifically, such as memory, and how important that is to assess and how challenging it might be to assess. And the possibility that we might dilute information by shrinking batteries down was brought up. However, that data has emerged about some promising correlations between the ways that measures are traditionally administered and CAT, again, with the caveat that these item banks are carefully constructed. So that being said, we've added some opportunities and hopefully stimulated some post-meeting professional collaborations that can come out of these. So discussions of stimulus or item generation via advances in AI were not discussed. I wonder whether we could just perhaps when we come back from the break, before we move on to point number 3, talk a little bit about AI. But for now, we'll take a break, 10 minutes or 9.5 minutes. Please be back at 12:45. All right. Thanks, everyone.
So hello and welcome. Benford and I are going to introduce ourselves, and then we will ask you to introduce yourself in chat, and we'll go through our hopes and our expectations for this breakout session. Thank you again for joining us. So Benford, why don't you go ahead and introduce yourself again?
Sure, thank you. Good afternoon, everyone. I'm Benford Williams at NIA. I'm in the Division of Neuroscience. I work in the Clinical Interventions and Diagnostics branch.
And I'm Elena Fazio. I'm also at the National Institute on Aging, and I'm in the Division of Behavioral and Social Research. And I just want to point out that we have one of our speakers from the first day, Dr. Siobhan Riley. She was our keynote speaker, so she's also with us. At this time, I'd love for the rest of you to go ahead and introduce yourself in chat. Tell us your name, where you are. And if you do decide to speak aloud, we'll ask you to introduce yourself, hi Siobhan, when you come on screen, if you so choose. So please go ahead and tell us who you are in chat throughout the time that we're together. I'd also like to point out Dr. Suzanne Hendricks, who's also a speaker in the Everyday Function group. We thank you for your participation, Dr. Riley and Dr. Hendricks. So as by way of reminder, we're here at the Everyday Function session. When we think about meaningful change in everyday function, we're really talking about the psychological, emotional, physical, and social function and how it can be understood and measured. So for example, we should consider what current measures are used to ascertain everyday functions, what the limitations of those measures might be, and who do they really help, and what opportunities do we have to perhaps further develop better measures and other considerations in the future. So I am going to open things up in just a moment after just sharing with you the goals of our time together. And we really do look forward to hearing from you and your input. So there are really three or four goals for today. The first is we want to hear from you. Many people didn't have a chance to speak aloud or participate in chat during the meeting. So we want to hear your perspective on everyday function. We're also going to respond to the 2 gaps and opportunities that you've seen throughout the meeting related to everyday function. So if you have any thoughts on those, we'd love to hear them. And then Benford and I are going to take some notes. And then when we go back to the fuller group, we'll share some key takeaways from our time together. So again, introduce yourself. And then I'm going to ask Benford now to go ahead and put up the gaps and opportunities slide. So we'd love to hear both your reflections, any additions to what you see on screen here that I'll read in just a minute, and any other points of interest and things that you want the group to understand. So we are responding first to these gaps and opportunities, and then we are delighted to hear everybody else's thoughts about what should be added to our discussion. So first, the first gap was really focusing on the development of approaches for multidisciplinary assessment of everyday function to include emotional, psychological, and social functioning that incorporates domains prioritized by those living with dementia, including maintenance of self, belonging, engagement in meaningful activities. The second gap and opportunity is really trying to focus on continued input from the broad community. So maybe the second one you could think of as being a little bit more focused on going to the community, with the community, and hearing their input on refining and applying approaches to outcomes measured for those people living with cognitive impairment or dementia. So I just kind of kept those at a high level. We'd love to hear from you. You can either come off mute and just speak whatever you'd like to say aloud, or you could raise your hand and we'll go in turn. So the first order of business is, does anybody have any comments or additions or changes for these gaps and opportunities, or do you have additional things you'd like to share? I see that.
Hi, I'm
Go
Sarah
ahead,
from
Sarah. Thanks, Sarah.
Alzheimer's Clinical Trials Consortium. So I sit in the room when we're designing clinical trials, and then I co-chair our participant advisory board. And I think what I'm seeing is this information isn't part of clinical trials. What is important, even the quality of life measures, if they are done, they're very minimal. And there really isn't that depth of information being gathered in the trials themselves. We're also, I think there's opportunities through digital and remote studies to gather much more rich information directly from our participants, qualitative information. Rarely do we survey trial participants to get this kind of information and their feedback. So I think there is, I don't disagree with anything on here, but I wonder if we can add more specifically that needs to happen within the trial space.
Thank you so much, Sarah. I was thinking of something similar and you put it in such a nice way. I think folks care about these particular outcomes related to everyday function, but just to underscore the point that you've already made, they don't necessarily make their way into clinical trials. And, I think we're talking a lot about drug trials, but we could also expand that, of course, to talk about dementia-focused trials that go well beyond drug trials. And maybe some folks have good measures that they know of in trials that are out there. And I know our speakers, and I see that Dr. Andrea Gilmore-Bakowski has joined us as well. She was a speaker in the Everyday Function session. I know there are studies that are trying to do more of this, but I think it's still a real gap, as Sarah, as you're mentioning. And I see that Cheryl, you have your hand up. Please go ahead.
Yeah, thank you. Hi, everyone. I'm Cheryl Kuhn. I'm a psychometrician by training, and I'm at the Critical Path Institute. Most of my work is specifically around clinical trials and constructing endpoints that are meaningful to patients, as well as the type of endpoint that regulators can make decisions on. So in the last 2 days, as I've heard these conversations, I felt that there was a bit of a gap between those two stakeholders. The things that are meaningful to patients might not necessarily be the things that regulators can make decisions on, because the decision has to be based specifically on the new therapy directly impacting that outcome. So things like quality of life that have been mentioned, absolutely critical. That's probably the first and foremost thing that might come to mind in the real world setting, but in a clinical trial, it's hard to make efficacy decisions based on quality of life. So as we're thinking about these approaches and thinking about the multidimensional domains that might need to be measured, we also need to think about, who is this information being generated for? Is it for a regulator who's deciding on a primary or secondary endpoint? Or is this information being generated so that once drugs do get approved, now the clinicians and patients have the information, and caregivers have the information that they need to decide if they should try a new therapy, or if they switch, not saying that right, should switch therapies. There we go. So I wanted to put that context into this discussion, especially because we're thinking clinical trials, but maybe things beyond clinical trials, and they're more academic research settings where the information does get used in a different way.
Wow, such a great point. I'm so glad you said that early on, Cheryl. The audience really matters. And I'm going to try to say this in a way that makes sense, but I know Benford and I, when we were talking about this session, we were talking about everyday function in what context. And I'm not sure that I can even put it as eloquently as others have, but it's everyday function within the context of a clinical trial. But I think what Cheryl's saying is, like, it's there, but it's also then well beyond there, right? Because, like, even just the language of everyday function doesn't engender thoughts of being in a clinical trial, because that doesn't really feel like your everyday experience, right? But at the same time, it would be nice, I think, to Sarah's initial thought, that we have different measures that actually do get taken up in the clinical trial process, so they become meaningful to those I don't always like to use the word patients, but I will hear patients and families to see what's of great value to them. So thank you both for those incredibly helpful comments. Andrea?
Hi, everybody. Yeah, I think what I also heard yesterday at the close was Luke mentioning that we also want to be inclusive of other types of trials. So we're also thinking about non-pharmacologic interventions and many other things that would be considered in the course of management and care for individuals living with dementia. So it sounds like we have a breadth of audiences. So I'm just confirming that that's true. We're not just talking about drugs, right? Does that sound okay? I'm seeing people not. So I have a list of many things, and I was going to share like just a few of things that I observed yesterday that I thought we talked about but didn't capture within these gaps and opportunities. One was the need for sensitive measures, and I think that works on both ends of the continuum. But when we're thinking about everyday function, we really don't have sufficiently valid and reliable measures of function at very early disease stages, and particularly for those thinking about the medication therapies, that's really problematic. So that seems like a potentially obvious opportunity to think about complex IADLs and other, and like maybe some of this comes through the digital measures as well. But that's a gap that I just think we mentioned but didn't talk about. And then I'm kind of hoping that Suzanne has ideas about this, but implicit to a lot of this is that priorities change over the course of living with the disease. And that, as well as everyone's trajectory and experience, is different. So it seems like there's a methodological need to understand ways that we can have sort of like more agile and pragmatic assessments that are reflective of where people are. And that's obviously difficult when we also want to compare. But I feel like we need to think about methods and models for flexibility in content and assessment strategy. And like we do this well clinically in other areas. So I think we can figure out how to scale that to research, but that seems like a methodological gap, like the group difference versus the individual difference. So you're on a fast slope or the moderate slope or the slow slope. It seems like your measurement needs to look different. So I'm curious if others have ideas about that, but we talked about it a lot. And I don't see that we've codified, what is the gap or opportunity with that. So I'm kind of suggesting that maybe we could talk about that.
Yeah, Andrea, that's a really good point, and it's one that I've spent a lot of time, and my team has spent a lot of time working on. If you take one of these scales, like an ADCS-ADL, and you assess what the most progressive items are in a pre-dementia population, a mild dementia population, a moderate dementia population, it's obviously very different things. There are different day-to-day tasks that are being lost in each of those different stages. And if you have a scale that is really broad, you can identify the most progressive aspects within each stage. And I think the first presentations on the first day that we're talking about cognition and talking about adaptive measurement of cognition, those same kinds of things can apply to function, where you could answer a few questions at the beginning and based on what you answer, go down a specific route. And I think it's really important to understand that those adaptive testing techniques allow you to know what is happening in the other scales, even if you're not asking those questions directly, basically by knowing that if someone can't do this, they also can't do that. And someone who can do this also can do that. And then, and really the order of loss of these things is fairly consistent across patients, but the speed of loss tends to differ quite a bit. And so all of that lends itself to this adaptive testing, having specific outcomes that are sensitive in different stages. And then the earlier conversation of really identifying instrumental ADLs that change very early. Some of the older scales don't have items that are really as relevant in those earlier stages, especially things that are related to using phones, using computers, things that weren't in the tests, 20 years ago, and we're still using those. And so I think all of that is critical. And then I do think also the quality of life layering on top of those kinds of measurements is also critical. And there are ways to bridge across the tests we use in research, the tests that are used more in the clinic, and sort of equate those kinds of things. But that's, we need the databases that have multiple assessments to be able to equate across things and then say, where are the gaps in each different stage of disease?
Having sort of this image in my mind of a matrix that's could kind of get really quite wild. Like it's almost like differences by audience, differences by stage of illness. I guess those are the two primary ones that come to mind. Anne, I see you've come on screen.
Yes, and I'm very interested in that piece, linking the quality of life to the different functional measures. So thank you for bringing that up. Added complexity there that I've seen with research participants is that some of those functions over time, not necessarily due to their cognitive decline, but that they recognize they can't fully participate in some of those functions, those functions become less important to them. So I don't know if it's how they're compartmentalizing the thinking around these functions, but in previous years, something was very important to them. Say, for example, driving. But now they'll say, no, that's not that important to me. So I think part of bridging that quality of life piece is figuring out what is the importance of each one of these as they might be changing over time with some of the research participants.
I thank you for raising that. I think that's really critically important, and we cannot, it has to be individualized on some level, and we have the technology to do that now. So we've been using such blunt instruments for so long. It would be incredible to be able to move this to the individual level, but still standardize it in a way so that it can be understood at a group level for regulators, and then for future, for future patients that are considering therapies, understanding what impact a therapy had on somebody's individual abilities. Because you're absolutely right, people do adapt, and even the fact that they're adapting could be something that's measured as an outcome, which currently is not measured in our studies.
I have a slide that might be interesting. Is it possible to share a slide?
Benford, can you take yours down for a moment and let Suzanne give it a try? Let's see what happens.
Okay. Okay, so this is just looking specifically at the research instruments that are often used and looking at all the different stages of disease and across which stages of disease these items. So all the ones in green are cognitive tests and where they're sensitive to progression. And then the ones in orange are global assessments. The ones in gray are the functional assessments. So we have ADCS, ADL, MCI, the ADCS, ADL, FAQ. The DAD could be included here too, and the Amsterdam IADL. And this kind of a thing could be adapted to include additional scales, such as quality of life scales and other types of instruments, and could just be expanded and going down. This is just an illustration of which stages they're relevant in, but you can actually, within each of these, also have things like which items are progressing and changing in each different stage. You can do little distributions of who's lost these things, or even those S curves, kind of like the Cliff Jack curves, where you're showing everyone can do it here. By this point, half the people can't, and by this point, almost everyone can't. And showing those curves side by side can give you a lot of insights into what tends to go together. And for some items, people tend to be very similar to each other. And then other items, there's a lot of diversity, and people, the quality of life may not connect as well with ADLs, or, specific items may have a lot more variability in them. And in some ways, the items that have more variability are less important for doing the group level things, but may still be very important for doing the person level. So, there's a lot of challenges, but I think just this kind of a map is a good place to start.
Thanks, Suzanne.
Sure.
And oh, go ahead, Tavon.
Sorry, I'm just trying to work out the technology, how to click everything. Really great point, Suzanne. And I'm just thinking, sort of layering on top of what you've just added, the sort of essence of what I was presenting on the first day is really in those, for example, quality of life measures. They need to be asking what matters most to people, because if these instruments don't tap into that, then we get to the situation where we're spending millions and millions of pounds here, dollars there, for what? We don't necessarily, they're particularly with psychosocial research, maybe drugs as well. Are we really actually measuring what is the drug or the intervention, what it's actually affecting? So I suppose that would be my caveat, really, is to make sure those instruments are validated for diverse populations as well. I think maybe that needs to be sort of added into the first researcher-based approaches as well. There's something about that, and the testing of measures. And I just think there's an incredible amount of work that is needed there. And I think certainly sort of what I've seen is that, we need to get the trials going. We need to do the research. And the impetus for that is so great that this work on validation of tools can just get left behind. It's so important, but it's not very exciting, really. It's just drudgery, sort of, in terms of sort of the exciting research, or compared to the exciting research of actually doing the research. But if they're not right, that fundamental building block is gone.
Siobhan, I have to say that if I remember correctly, yesterday you were talking about the fact that if we're not measuring things appropriately, we may be missing the mark on things that are actually working. I think that was you, Siobhan.
Yeah. Yeah.
And so I'm thinking about, this is the preaching to the choir, so to speak. This group is folks who are selecting into a meeting about measurement and then selecting into this discussion. How do we get researchers to get more engaged or interested in the really important topic of measurement development? Many of you have been doing this work for a while, and you may say, it's a long road, but I think that point you made, Siobhan, about folks who are running trials might be missing the mark because they're actually doing something that's really quite efficacious, but we're not picking up on it. How do we sort of motivate around that? Because it does seem infinitely important. And I think thinking back to your talk as well, that if we can help researchers to appreciate that it's better research if you have better measures, and you're also hearing from the people who really matter and who really care about these outcomes, maybe those dual purposes could be useful. Suzanne?
Oh, you're on mute.
I'll go off mute first and then talk. I think one of the challenges is that there are, when you look at individual questions and items across all these different types of measures, and so you're looking at, functional tasks, cognitive performance, just individual items, there are items that are really sort of markers of the disease, meaning that they're progressive, they're consistent across multiple people, they're really defining the progression, and you're able to use them as metrics of how fast the underlying pathology is actually progressing because you're seeing it on those markers. And then there are other items that are more individual and fluctuate and come and go on a day-to-day basis and may still be very important to people, but those are much harder to change on a group level because individual people will be seeing, this is how it shows up in my life. This is how it shows up in someone else's life, and it may be completely different items. Those ones that sort of come and go and aren't really common across the whole population and are not necessarily indicators of the underlying disease progression are different, and they're much harder to manage in terms of being able to measure them clinically, being able to change them on a group level with any kind of intervention. And I'm hopeful, and I believe that those kinds of fluctuating ones that really are person specific are driven by some of the underlying things, and they just impact people differently. And so as a researcher, and a researcher who's really focused very strongly on efficiency of clinical trial analysis and really making sure that we're getting the most we can out of these studies, that we're giving the treatments the very best chance of succeeding. We really focus on those progression markers that are more markers of disease than necessarily individual customized features. And in that process, as we measure also those individualized customized features, I'm hopeful that those will show similar changes. But they're things that can't necessarily be part of the core metrics of how you measure a treatment effect because they're not common across everyone. And so I kind of think that we need to divide the conversation into those two different kinds of outcomes and make sure we're addressing both. And I think there are a lot that are key features of disease progression that are also very important to individuals. And they can say, yeah, these are important to us, and we can, match those up and see that. But I think there will be some that are very important to people and are not necessarily progression markers. And it does add a lot of complexity.
That's a great point. And as you're talking, Suzanne, about like kind of setting into two different topic areas, I want to just go back to something. I don't know if Dr. Meny feels comfortable coming on screen or articulating her comment. So I'll just maybe lead her in that direction. But in chat, she talked about the interest in having common measures across different audiences. Heather, I wanted to ask you to say a little bit more about that, if you're willing.
Sorry, I was finishing up some lunch really quick. Part of what I've been thinking about for a number of years now is kind of, I want to use the word harmonization, but I want to make sure we're all on the same page as to even what that means. And I think Benford commented also, like, the different audiences. So like right now, a lot of what we're talking about is clinically meaningful. And we're talking about in terms of trials. I think of that for pharmacological and non-pharmacological. I think of that in terms of taking a step further to this new model CMS is rolling out, the guide model to help support people with dementia. I think about that in terms of the community-based organizations who are doing services. As Benford mentions, like, do we all mean the same thing when we think of everyday function? My background is not on dementia, decision-making capacity, but on decision-making involvement. Do we all kind of view that concept as the same thing? Do we all view caregiver burden as the same thing? And how is that useful moving forward? That's an even bigger issue, but just want to throw that out there to see if there's any added reaction to that.
Andrea, before you go ahead, Heather, it's great. I'm, nerding out here. It's great to hear people speak out loud and extend your thought, because what I thought was different than what you said. So where my mind was going was more of along the lines of, dare I say, sort of like a minimum data set, meaning like, are there certain measures that we'd want to have in common across studies, and then have certain measures or indicators that are different, so that you might be able to pick up on some of that individual difference that we're most interested in. So I thought you were talking about like having certain common measures across audiences, and that's where my mind went. I don't know if that resonates at all with what you're thinking or is in a different direction.
No, it absolutely resonates with what I was thinking. I think I was just spinning further on the comment that was made in the chat about we would also need to get agreement as to what that minimum data set and those concepts involve. I think the work by Dr. Riley, the work that was also mentioned by Dr. De Benedetti, starts to point us as to what is the meaningful piece for families and individuals. And I feel that's where we need to start from. But that just to have the ongoing conversation to push us further, a minimum data set would be magical for so many reasons, for so many people. Not the least of which is improving care and services for this population.
Thanks, Heather. And Andrea, I see you put your hand down, but I didn't mean to ignore you. Do you have
No, I had something about Siobhan's earlier comment, but I also really appreciate this comment. And someone, I think Emily made a comment in the chat about also like the detail that can be acquired with some of these outcomes. Like, someone may experience, like if we think about non-cognitive symptoms, maybe they experience agitation, but maybe it doesn't bother them and it's not disruptive. So it's a very different experience. And this just made me think again about the fact that we want to be thinking about non-pharmacologic interventions as well as drug interventions, and they have different mechanisms and they target different targets and they should produce different outcomes. So we should be aiming to do something slightly different, right? If I'm going to provide someone with an individualized, meaningful, engaging activity, maybe what I want to do is promote engagement. And I don't know that that's the goal if I'm treating someone with an anti-amyloid therapy. So I do like the idea of like a minimum, core outcome set for things that are important across stages. And then I also think sensitive outcomes is about sensitive to what, so that selection is still important. I just wanted to make a quick comment about, I completely agree that we don't, and I did kind of want to mention this yesterday, there is a field of positive outcome measurement in dementia for like at least 20 years now. And so it's not really as novel of a concept as I think it sounds like here, but I do think that field could make a lot more progress scientifically and could be brought into this mainstream. But a real challenge is it's very difficult to fund outcome measure development. The Alzheimer's Association is maybe the biggest funder of this right now they have this ARCOM initiative where there have been funding with the Link AD, sort of sensitive, meaningful outcome measures. So I think that's a big challenge. But the other is we really have a gap, which is, and I'm trying to do this right now with some emergency care, person-centered outcome measures. We don't have like a sufficient toolbox of how do I do cognitive debriefing and how do I validate measures, you know, with individuals who are experiencing cognitive symptoms who can absolutely tell me what matters to them in the emergency department. But I don't know the best strategy to follow what the field would like to see for the gold standard for validation of that through like the cognitive interviewing and the cognitive debriefing, which are very cognitively challenging tasks anyways. So I feel like that's a very specific methodological gap that would support what Siobhan is mentioning, which is we need to develop the measures for things that matter to people. And then we need to the best practice methods wise for how we should do that. And I don't see that that's been developed yet.
Siobhan, you pulled Andrea, you took your hand down. Go ahead.
Thank you. Well, yeah, just to pick up on Andrea's point again about it is really hard to get this sort of work funded. And I think there are often caps as well. So even if there is a funding program, the amount of money that's available is probably not really enough to do a program of work and to work across a lot of researchers. So one of the things that I alluded to in my presentation was really about we need to start some way of working together as researchers on this, because it doesn't necessarily take a single study that's focused on this. It could be embedded, it could be a study within a study. And again, it's, you know, if people are going for funding, then it could be a work program that is focused on developing or testing a measure within a different population. So I think there are just lots of opportunities that may be missed because maybe we didn't start something like this 20 years ago. So this is an amazing opportunity to be able to even be talking about this because it's so important. So yeah, sorry, I'm just wittering now.
No, not. And actually, this is a comment more than any, not a question, but for those who didn't hear me introduce myself, I'm at the National Institute on Aging, and we and some of you on the call know that we have an Alzheimer's and related dementias network that's focused on home and community-based services measurement, among other things. They do more than just that. But I mention that because I know that there's a lot of my colleagues who try to develop measurement-oriented research networks, right? Like that you get funding for. But one of the things that comes up for me a lot, and I use the home and community-based services one as an example, is we end up in sort of these different lanes and pockets. You know, it's like so we're having similar conversations, but not all together. So again, it's more of a comment than anything else as I'm thinking in my head about like how do we move the field or fields plural forward? Like Siobhan, like you're saying, like this is a great start. It's slightly tangential than some of the networks that I know that already exist. And so just thinking about how we might get folks together in a meaningful way. Like this meeting is specifically about clinical trials, where like the network I just mentioned, it could be related to clinical trials, but is often not when we're talking about home and community-based services for those with dementia. So just really a comment and sort of reflecting back to you, what I'm hearing is a concern about how we move things forward, and also acknowledging Andrea's point that lots of people have been doing work in these areas for a long time. I know a number of you have. This is not new, but like, how do we continue to advance to kind of ultimately get measures into the right places, into trials, into the correct audiences with some consistency? Yeah, I don't have an answer to that, but I'm just putting it out there. Suzanne?
I don't have an answer either, but some additional thoughts. So I think coming back to some of the comments Andrea and Siobhan made, there's a very dramatic difference between this sort of progressive disease modifying kind of concept and symptomatic concepts. And we work in neurodegeneration. We also work in psychiatry, and a lot of the psychiatric outcomes are very much symptomatic and moment by moment. And those are very important symptoms, but the same the same way of identifying the best measures doesn't work for psychiatric measures as for like the progressive aspects of Alzheimer's disease. And so I think when Andrea asked, okay, what do we need these to be sensitive to? The answer ultimately is treatment effects that would be beneficial for individuals with the disease. And if it's a progressive disease and you want to measure the most progressive parts and you think you're slowing down that progression in some disease modifying way, there are really good ways of identifying the most progressive aspects of disease. And right now we have pretty good assessments. There's probably not a lot of room for improvement in the mild or later stages, but when you go earlier, there's a lot of room for improvement. And there's an objective way to measure how good these assessments are. And it's just measuring the signal to noise ratio or the mean to standard deviation ratio of the change scores over time, because that's really a surrogate for time. You're really saying how much time has passed, how far in the disease are they, have they moved farther along in the disease. Now, on top of that, and we do need to go earlier. We need some of those really sensitive outcomes that are progressing in the very earliest stages where you can start to see functional changes. And I do think some of the digital markers will help us in those early stages, but even questions that are related to really more complicated tasks that are being lost very early. But the second piece of that is the symptomatic component, and the behavioral outcomes will likely fall into that because psychiatric and behavioral end up being very similar, and because it's neuropsychiatric outcomes. And those ones are things where you can get improvements over your baseline, not where you're just slowing down progression. And in that case, you're really looking for sensitivity to the specific kinds of treatment effects you expect with the interventions that you're making, whether they're lifestyle, nutritional, or whether they're drugs. And again, in the very early stages of disease, we haven't really pulled over some of the outcomes that could maybe be more relevant in those early stages. Some of the neuropsychiatric changes that happen really early are agitation, anxiety, depression, and we're not really emphasizing those in the earlier stages in clinical trials right now. And we probably are seeing changes there. And there are really good tests that are used in psychiatry that could be pulled over to be used in that stage. And then I think what would be really interesting is to line that up with research that's already been done, asking, doing interviews and saying what's important to you in terms of changes in different stages of disease, and see if we already have things that are measuring that, and then see if there are new pieces, gaps that we have, and new things that need to be added. So I think it's really important to do, a full assessment of what's already there, see how well it's performing for either progression or these symptomatic types of changes, and then find the gaps in those that are really not being measured, and then fill those gaps rather than sort of starting from scratch and doing brand new work. Take what's already there, find the gaps, and then see the best way to fill those.
If others don't have continued thoughts based on the conversation we were just having, I'm curious if we could go back to, I think it was Heather's point again about how we actually define everyday function, just maybe for a bit of a thought exercise here. What are the different ways that people might imagine everyday function, right? So like, for example, when Benford and I were thinking about this, we were talking a lot about IADLs and ADLs, and that seems to be like where most of the conversation resided. But I feel I have the sense for a number of you, everyday function might go well beyond the bounds of instrumental activities of daily living and ADLs. I'm just curious if anybody could comment on how you conceptualize and think about everyday function, and if it's more expansive than that, I'd love to hear how you're really thinking about it. And I will just also add to that, if you have something that's in more of the emotional, psychological frame, what do you think of as the overlay between everyday function and those things? Like, is it anxiety and depression, as was just mentioned, or is it beyond there? So thoughts on how we actually are conceptualizing everyday function broadly, Sarah?
Yeah, thank you, Elena. I think what we hear from our participants, so instrumental activities of daily living are things like, fixing yourself, going grocery shopping, fixing food for yourself. But it's the social interactions and the ability to participate in social engagement that seems to be emerging from a number of spaces that is important to participants, plus it's what we're hearing directly from our participants on. So I think there is more work to do there. I think there, especially going to those earlier stages of disease, eventually we want to be running prevention studies, early preclinical studies. So maintaining the activities that are most important to you usually are very complex tasks that include social engagement, picking up my grandkids from school, being able to function on my own, continuing my volunteer activities and work.
Thanks, Sarah. Anne?
Yeah, I would add decision making. Many of our research participants will voice that they're being left out of decision making. So I would add that to the other social list.
As you all are giving these examples, I'm thinking again of our conversation earlier about audiences and sort of like measures that might matter to one particular group or another. So I think what we're talking about in a very important way are measures or outcomes that matter to people in the lived experience of, in a trial or outside of a trial. And as you were saying, Sarah, like if we're talking about an earlier stage of not even illness, but just prevention, that like across the lifespan, people care about the wide array of quality of life and social engagement measures and mattering to people, other people and purpose, whatever. So I think what I'm hearing the group talking about is going back and across the lifetime, the life course, and thinking about the things that we care about in terms of a broad conceptualization of function and what matters to us as being important to people throughout the whole progression of illness, but it'll change a bit. And then we also have measures that are going to be of interest to an audience that's outside of that, right? So if we're thinking about a payer or a regulator or someone providing a service, they may be thinking about the measures in a slightly different way, and maybe it calls us to try to capture. The answer is always capture more data, right? Like capture more information about people. But I see that a couple other people came on screen. Does anybody want to add to that?
I haven't been able to attend the last three days fully, but I enjoyed Dr. Riley's presentation on the very first day. And I'm wondering if you have easy access to your slides. Because I remember there being a very nice summative slide of the concepts. Maybe there's some nice pictures on there too. I feel like that may also, Elena, like help.
Yeah, I can
do
do
some
that.
added discussion on this.
I was just looking at them and trying to just think this is everyday function. So I'll just see if I can share.
Yeah. Oh, great. Oh, I see something changing. Nope.
Well, it's just yeah. I think this is going to work.
Okay, good.
So let me just see if I can. Oops, wrong one. Anyway, if I make that bigger there. Can you see
communication. That's one we haven't dove into quite as much yet. Communication factor, and that's not part of the typical ADL, IDL piece. That's nice. Thank you.
And I think the feeling able to have a laugh with other people. I think it was interesting. Was it David Seller? Was that his name? Who talked yesterday about the NIH toolbox, and a lot of these items just weren't coming up. I think he said he managed to find five or measures that would capture 5 of them.
Yeah.
But this sort of feeling valued and respected by others really came up in the first session today, and all the discussion. But it's just not in the measures. It's just not something that we currently measure very easily. So yeah, to let me know when I should stop sharing. But thank you for raising that again.
Thanks to you both. I think you can probably leave it there for now, Siobhan. We only have, I think, a few more minutes before we go back to the fuller group. And I will try my best to hit some of the highlights about what we talked about here. I've been taking some notes, and I know Benford has as well. And I appreciate this particular slide, and then your mention and Heather's as well about the promise overlay that David Sella did. And I think I remembered even a smaller number than 5 of the items, but maybe that was right. Maybe maybe
I joined really late. I apologize. I've been traveling today, but yeah, it was 5 out of the 13.
Okay.
Yeah.
Got it. So I think, David, thanks so much for joining us. We've hit on a lot of different topics in our discussion here, and I'm wondering if I'm going to try to highlight some of them when we come back to the fuller group, but maybe we could just take the moment or two that we have left. And I'd love for people, if you had a particular piece of our conversation that you think would be great for me to highlight as we go back to the fuller group, please vocalize that. That would be helpful.
I think that talking about the distinction between clinical trials and everyday practice versus even observational trials, I think that's critical. I think that the sort of immediate and unique symptoms or experiences versus the more common and progressive ones, I think that's an important concept and came up in a lot of different ways. And then this idea of taking the best of what we have in the stages where we have it, but moving earlier with better measures and identifying really the gaps based on what we see, because I think most of the gaps will be in the earlier stages. And then I guess a fourth one I would think based on, what's on the screen and then just the conversation toward the end is being broader about our concept of what everyday function means and making sure it's not just ADLs and tasks and things, but it's all of these concepts and the emotional and psychiatric, psychological relationship aspects as well. And even cognition in the way that it plays into day-to-day tasks and day-to-day life. That was my sort of summary. I don't know if anyone else, if I missed anything, I probably missed some stuff.
I would, that's wonderful. I think, I keep going back to this notion of audiences and what changes over the progression, what we can capture over the progression of illnesses as well, over the progression of illness, and how I'm just mentioning the part that you just said, Suzanne, about the gaps may be unevenly distributed. We're leaving the breakup room. Okay, according to where you are in the disease progression, that we may have more ability to measure it in one part of the time frame than another. And I'm just going to go back and try to grab any of my additional notes. And then Andrea.
I'm just agreeing with you. I think that's really important because we didn't get to it the last 2 days. And I think the other thing is we're talking about measuring what people have, not just what they've lost. And I don't know if that's being discussed in the other sessions, but that's another way to kind of frame these other things we're talking about, including.
I heard someone say the thing you're about to lose and the thing you just lost are the most important things to you at the moment.
You were, I thought that was you who said that. Okay, it wasn't you. Okay. Another wise person. Okay, thanks everybody. We're heading out in 2 seconds.
With just a quick round of introductions. I'm Chris Weber, Director of Global Science Initiatives with the Alzheimer's Association. And was, for those of you that may not have been on the session yesterday, was moderator for session 3, the biomarkers session. So happy to be leading this breakout session now. And yeah, well, I guess we'll get started in a minute here. I think the next thing I'll show is the session 3 gaps and opportunities slide. So we'll get to that in a moment. So Amber, Christina?
Yeah, sure, I'll go. My name is Amber McCartney. I'm, am I unmuted? Yeah, okay. I'm a project manager at NINDS, and I work with the Alzheimer's disease and Alzheimer's disease related dementia program. And as part of the planning committee for the workshop and the biomarker session, I will do lots of listening today and take notes. And I'm really, grateful for the opportunity to hear everybody's voices here today. Christina.
My name is Christina McClendon. I am with the National Institute on Aging in the Division of Neuroscience. And I oversee a portfolio of non-pharmacological clinical trials focused on the person living with Alzheimer's disease and related dementias. This has been an exciting set of days so far, and I'm looking forward to today's discussion. Thanks.
Oh, you muted.
Chris,
Sorry,
I think
there
you're
we go.
muted.
Now I'm unmuted. What I was just going to say, thank you very much. And I think now what we'll do, we've got a great crowd now for this discussion. So it would have been nice to have a slide for this next little series that I'm going to go through, just to sort of let everybody know what our format is, what we're going to be talking about here. So I guess our goal over the next, what is it, next 45 minutes, hour that we have, we want to, our goals here are to hear from as many of you as possible, especially if you didn't have a chance to ask questions or voice your input during the last couple of days of the sessions. And I think our gaps and opportunities slide is a great first step here. That's a good starting point to see if we have any other input to add. We won't be editing the slide in real time, but I think all input that we get on these gaps and opportunities, I think, would be very helpful to our NIA colleagues. And we will be, I think, Amber, as you said, we're going to be taking notes based on this input and this discussion, and we would want to share back with the larger group after the breakout. So, so I think, yeah, that's all I wanted to say, just to kind of tee up the conversation. Our goal here is to discuss some of these priorities. I think there was a lot of great input during this session in particular. So I thank everybody for taking the time to choose this session, which is really important, both from a research and a clinical perspective of where we're going with biomarkers and clinical meaningfulness. So with that, Amber or Christina, do you have anything else to add to sort of tee up our conversation here?
I'll just say what Christina put in the chat. It'd be great if people would we would appreciate knowing who you is here, and if you have the opportunity to introduce yourself in the chat, would be great. And I think also, like, cameras on, and just we're using the hand raise feature. Just wanted to ask that. Would all of that, like, just so we can, have a lot of back and forth and make an easy conversation kind of style would be great. You don't have to just put your formal comments and questions into the chat. Like, want to have a conversation. Thanks.
Totally agree. And maybe the first time you have your camera on and make a comment, it'd be great just to maybe even verbally just say who you are, what your affiliation is. I think that would help orient everybody too.
Fanny, I see your hand is raised.
Yeah, thanks, Christina. My name is Fanny Alahi, and I spoke at the biomarker session. I work on new biomarker development, really focused on developing new biomarkers of microvascular disease. I wanted to maybe bring back the discussion around amyloid, because I always say amyloid may be part of some people's disease and not all. And then at the same time, and I see that Ron Peterson is here, and his cohort would be a really good cohort for this, is at the same time, I wonder whether we're just looking too late for a lot of people, and that if we looked at an earlier stage, we would find a stronger association between amyloid in many more people and the clinical outcomes. Again, not everyone, because AD neuropathology in of itself may not be a big component of a lot of people's decline, but the timing is a big part of why things show association, I think, with clinical outcomes or not. And because of who we study and how we study people, I think we fail to find the earliest drivers of pathology. So we'd love to hear thoughts on that. Hi, Rod. I see Rod Corvo here as well.
Hi. Hey, I could comment if you like.
Sounds good.
Hey, so I'd like the kind of the pivot that you made at the end of what you just said, Fanny, because you pivoted away from association to driver. And for me, that's the conflation of biomarkers with causes without, I'll say without full evidence is kind of a framing challenge that I think has not been overcome. So I don't know if how much going earlier is going to help, but I like the idea of pivoting, like recognizing the value of associations, because there is a lot of value there. Certainly, we're looking for that in Mark VCID, but also being cognizant of the need for that pivot. So I'll just stop with that.
I can say a couple words. Ron Peterson at the Mayo Clinic, and thanks, Fanny, for calling me out on this. You're right. I think that most of, at least in the amyloid world, and again, that's not the whole world by any means, the indications are perhaps the earlier we intervene, the better. Certainly some of the more recent data from the clinical trials, the Clarity and Trailblazer, have indicated that people with the least amount of, say, tau pathology in conjunction with their amyloid pathology may be the best responders. And so that supports your notion of going earlier. Again, amyloid and tau are not the end of the world. So as Rod is saying, we really need to look at the earlier and earlier indications of microvascular disease that may be evolving. Is it association? Is it cause and effect? Hard to know. But I think getting this information from longitudinal populations that are normally aging, so to speak, to gather some feel for how these various biomarkers evolve over time, and then hopefully we can sort out which is an association and which is a cause.
Can I ask you a question, Ron, maybe going back to what, and anyone actually who would like to jump in, what you said, Rod, about causality. So in the back of my mind, and sometimes I voice this, but most of the time it's quiet thought, that our anti-amyloid therapeutics effect is actually not the clearance of amyloid per se, but something like immune activation in the brain, modification of factors that we're not directly quantifying with biomarkers, amyloid being a really fantastic biomarker of so many different processes, clearance being one of them, and that we are actually helping the brain's clearance system. All we're looking at is the clearance of amyloid, but there may be much more toxic things that are getting cleared and or cellular function restored. So yeah. And there's something to be learned, right? Because the only causality that we can go for is when we're intervening with clinical trials in humans. And mouse models are falling short of capturing Alzheimer's disease complexity. So what are your thoughts about the beneficial unknown off-target effects of the anti-amyloid therapeutics? And how could we capture that in a meaningful way, actually learn from it?
Rod, is that you or me?
I'm looking at you.
Well, I can just say that no doubt. I mean, if you look at the mechanisms, the putative mechanisms by which the, say, anti-amyloid antibodies work, it is activating an immune mechanism, activating microglia that actually clear the amyloid protein, but probably clear a bunch of other stuff too. Certainly dystrophic neurites, maybe some other inflammatory processes that are happening. So you almost think if you could activate something like a lysosomal system that would really clear a lot of multiple toxic proteins, that could have a beneficial effect. Someone said yesterday, what I've always thought about is that we study amyloid so much because the light shines more brightly there. In other words, we can measure it. We've been able to measure it in the spinal fluid for decades. We can see it on PET scans, and now possibly in the plasma as well. So we just are able to study it more closely. That doesn't mean it's the end all be all. It just means that it's received a lot of attention. We should expand that to the other proteins as well.
That's a great point, Ron. And just to add to that, one of the things that the Alzheimer's Association does is fund the full spectrum of research, precisely because of what you're saying. Amyloid probably isn't the answer. At least the drugs that we have right now are not the answer to cure the disease, but they have shown an ability to slow the disease. And only 20% of the association's hundreds of millions of dollars that are invested in research is solely amyloid focused. There's so many other targets out there that I think we need to learn about to know what their association is, what the driver is of the disease. And I think the more we understand the biology of what's actually happening well before clinical symptoms are present, before you see the memory and or functional loss, we need to understand what's happening with all of these other targets. We're
Chris,
building. Yeah, go ahead.
Can I turn for a second to the first point on the slide, especially in light of the discussion we just had from persons who are involved with these diseases, is that what I was hearing from them is they really want return of result. And so that pertains to number one here, returning results of biomarkers to individuals. But when we're dealing with underrepresented groups that have been inadequately studied, how do we give these results back? What do they mean? If you have an elevated amyloid, an elevated GFAP, what does that mean for an individual when we're talking about understudied groups? So I'd be interested in what the group has to say about how we can, one, give these results back because people deserve them, but we have to couch them in reality as to what the implications are of these biomarkers.
Excellent question. Anybody want to chime in on this?
I actually would like to chime in on that.
Was that Barbara? Go ahead. Yes,
This is
go
Barbara.
ahead.
Yeah, Barbara Bolstead from Madison, Wisconsin, and I'm in the research study with ADRD here in Madison, UW Research. About a year ago, I'm pre-diabetic and have that in my family, and so I wanted to do something about that. And so I joined an MA, Medicare Advantage, had a program called Quartz, which had a VERTA program to help with reducing obesity and reduction of my A1C, which had crept up to like a 6.0. I mean, it wasn't like 8 or anything, but I was still concerned. And because of my information about Alzheimer's and dementia and the importance of, maintaining a good weight and maintaining a healthy, overall, my health, exercise, and all that, nutrition, I focused on that. Well, I do daily biomarker. I do a daily blood glucose reading, actually a couple times a day. And I've been doing it for the last year. And I'm able to see the information about when it goes up, my biomarkers, my blood glucose, and something like that. I actually have a coach that if I have a concern about something for the day, I get my weight, I take my blood pressure, and I do this on a daily basis. I've lost 15 pounds just by change of diet. I'm not on medication other than my hypertensive medication. And that has really encouraged me to stay the course. So I say that because something like that, and I'm just thinking in terms of if I got sort of immediate response or immediate results from what I'm doing, that I'm heading on the right track, I'm doing the things I'm supposed to do. That really did change my behavior. And then I'm able to, if I have a question, text the coach and let her know, wait a minute, I was away for the weekend and this happened, I maybe overate or something and my blood pressure went up. And so what do you think? Well, we just watch it, drink more water. I know the things to do, but having someone on the other end who's concerned enough about me and who knows me is very helpful. So I don't know how that can, but just as a context for what was valuable to me, having something like that where changes, things that I have questions about, I don't have to go to a clinic or wait for an appointment. I can almost have something that's immediate but it doesn't put the researchers and folks more time out. It's something where you have someone monitor, the responses, monitor the information, and then if there's need to, it goes up the chain to someone who maybe has more information about that. I don't know if that makes sense, but that's just a thought I had, because I was focused on diabetes, and yet I know it's helping with all the other things to prevent dementia that I can do. So I know it's all related, and it helps me because diabetes in my community, people understand. They have a respect for it, a concern for it, about it, and so people understand that. And the tau and amyloid and plaque and all the other things, they may not, but they're related. I think, as that was mentioned by Dr. Fanny, that it's related, and being able to relate those to people that may not follow all this other stuff, that we're talking about, is going to be the way to go. I think that's just my input.
That's a great
I think that's
point.
particularly valuable for you, Barbara, and that was an excellent point, and it actually impacted your medical care. What I'm concerned about, and I think you may have mentioned earlier this morning, is that an amyloid feedback to you may or may not be as important in say a person of color versus a white person, because all the data have been acquired on basically white individuals. So we don't know the meaning of an elevated amyloid in other populations. So I'm concerned about giving back erroneous information or information that we may not be able to interpret properly, but that's where we need more longitudinal data. Studies like you're in, the RAP study is a perfect one to gain longitudinal information on the role of these biomarkers. So I agree completely with what you say, that it impacts your health, and that's important. We need to acquire the same kind of meaningfulness for our other biomarkers.
Oh, Barbara, you're muted. And after that, I see Nancy has her hand raised.
Just saying thank you for that. I totally agree and appreciate, the understanding of what I was trying to say. Thank you.
Go ahead, Nancy.
Thank you. I introduced myself in the chat. I am in my third year in the AHEAD 45 study of lecanemab. I found out 10 years ago that I had ApoE44, 2 copies from 23andMe, which is not the preferred way I would recommend someone doing that. And found out I had elevated amyloid in 2020 at the end of the Generation 1 trial. Through a very brief conversation, because it was right as COVID was shutting everything down. So again, not the preferred way of disclosing that, but I am a strong advocate for disclosure. I actually just last week had an infusion and have a great relationship with the study coordinator in my wonderful site. We talk about how screening is going and the quotas and the science. And she said out of 200, I think it was 221 people who screened for AHEAD 3 or 4 or 5, only about 10% have actually enrolled, but only 2 people chose not to learn their ApoE4 status. One of whom said she chose not to learn that simply because she had a good friend whose mother had a very long, difficult journey with Alzheimer's, and she found out she had ApoE4, and she couldn't recover from it. So this person was worried. But the vast majority of people want to know this information. And I actually know of people who did not qualify for the AHEAD 3 study because they were not at the 20 centiloid mark. And their response was, these actually were both professionals who said, well, how close am I? How soon will I be there? I want to know more. And I found myself in the not, sort of, look, I'm not a medical expert, but these things are uncertain. And I don't think, as much as we all would like to know that, anybody can tell you that. So they're not being mean and they don't tell you this. But I think we infantilize people who are clinical trial participants, whether they have a diagnosis or not, when we assume that they can't understand levels of uncertainty, and that it's helpful to, quote Sid O'Brien, to say, it appears that in the African-American community, or in some, but not all, Hispanic Latino communities, this is less relevant, but we wish we knew more. I feel like, as someone who was sort of gobsmacked with the term preclinical Alzheimer's disease 4 years ago, that having someone say, this is what this means, and it's real uncertain whether the drug I may or may not be taking will have an effect. And just sort of a final comment, Jason Karlowicz is someone who I really respect. I'm a graduate of Penn myself from a very long time ago. But I have been bothered recently when I've read that he says amyloid is not ready for prime time and we should not be using this in clinics and we shouldn't be sharing this and we shouldn't be making decisions about treatment with this. And I feel like saying, easy for you to say because you're not facing the train having left the station potentially. Like, your values are important. My values and my goals are at least as important. So I wish I could learn my tau results. I wish I knew more about my biomarkers. I actually one more thought. I have been told just within the last 6 months that there's some discrepancy between the local read of my MRI results, which was no ARIA-E or ARIA-H, and the central read, which is 3 possible microhemorrhages isolated, which could happen because of my 4/4 status. I value the fact that my study site doctor spent a good 20 minutes talking about what that might mean. So I'll stop there. Thank you.
Excellent points, Nancy. And you're actually, one of the things that you said too that I think really resonates is my goals are just as important. And they are. And hearing from folks that are in research and what is important is very important for the researchers to hear. Barry.
I don't want to dominate it, but I can just reflect a little bit on what Nancy was saying, and in particular with regard to Jason Karlowicz's comments. I suspect those were from a quotation he recently put out regarding the new Alzheimer's Association Alzheimer's disease criteria that are being developed and will be published very soon, that emphasize that maybe amyloidosis in and of itself is sufficient for the diagnosis of Alzheimer's disease. There's some subtleties in that, but I think that's where Jason was going with that. But I agree with you, Nancy. I mean, if that's information that you would like to have and can interpret it properly, meaning we can help you interpret it properly, I think is where the field needs to go. But it's that I think gap between what does this mean for me versus what does this mean for a large group of people like me. That's where the challenges come.
And just a quick comment on that, Dr. Peterson, is I am a moderator on a forum that is not very encouraging of mainstream science, put that way, on about 8.4. And so comments that are, and I actually read his article, so I know it's about the diagnosis and should it be A positive, T positive, N positive. But that gets taken out of context for people to say, see, even this person doesn't believe amyloid is important. So you can't change how someone distorts your views. But I think I love the idea of trying to change the conversation to amyloid may be a proxy for many important things, because people do understand terms like inflammation and they might take that over, well, who cares about plaques? Thanks.
Thanks, Nancy. Barry, and then John.
Hi, thanks for recognizing my hand. I have a few things to bring together here to this discussion. First of all, I want to thank the people who were in the previous panel. That was a compelling panel, and I appreciate the fact that some of you are participating in this discussion. I'm at Johns Hopkins School of Medicine. I'm also the editor of the Alzheimer's and Dementia Journal, Translational Research and Clinical Interventions. And for some of you in this group have already heard me talk probably ad nauseum on the importance of inclusion science, which is one of our special topics for the journal that I edit. And that's a way of rethinking diversity, equity, and inclusion. and the importance of understanding how biomarkers and clinical outcomes vary among racial and ethnic groups, people of different ancestry, if I can be a bit more accurate. And I've often challenged my colleagues in the past, especially when I've been in the privileged position of a group leadership, to please try to understand the difference between what we know and what we merely think we know. And so looking at the approval of lecanemab, I take that as an example. When this came out, a group of my colleagues at the journal, as well as colleagues outside of the editorial board, put together an editorial that was published last October 10th in the journal about, not commenting on whether or not it was a good or a bad thing to approve the drug. It's approved. It's reality. I have my personal feelings about it, and I'll betray the fact that I'm fairly positive about it, and I think there's some important advances that will grow out of these research studies. But the editorial highlighted the fact that it was a homogeneous group, basically, that was tested, this drug was tested on. We understand the effects, we understand the side effects in a very limited section of the population. And if it's going to be made available by FDA approval, then we need to fully inform patients who might choose to expose themselves to this drug of what we actually know about this drug, who was tested, and if I'm, I have thanked the pharmaceutical companies in the past for being so concerned with people who look like me, white American of European descent. Thank you. You've studied me very well. But what about the other people who may need to take these drugs who have not been studied yet? We don't know. We don't know what the significance of the biomarkers are, of the clinical outcomes are, which are the clinical tests, the cognitive tests also have biases to them. And so patients need to be fully informed before they make the decision of what they want to undertake. And that includes things like expense and inconvenience. And if my dog barks in a minute, please excuse me. I'm enjoying a spring day in the backyard. So that's one part of the story. The other part of the story is we have to embrace what we do know and get as much information out of the available data that we can. So, what do these biomarkers mean? I thought it was a brilliant question. Are these drugs really acting because of their impact on amyloid clearance? Or are there other mechanisms that are intersecting with the clearance of amyloid that may actually be impacting on the clinical outcomes, the clinical effects? So if we look at a clinical trial outcome, we have a treatment arm, we have a placebo arm, and these are large numbers of patients. And what we're looking at is a distributed measure across everyone who's in that treatment arm. So everybody who is on drug, there's a line that's drawn. Everybody who's on placebo, there's a line that's drawn, but it does not reveal the fact that there are going to be variable responses within those groups of individuals. So when we say there was a 27% slowing in the rate of the CDR, well, what do we really know? I mean, that's the group mean. That doesn't account for people who may actually have responded better or may have responded less well. And what can we learn from? I mean, if you look at what is accumulated in a clinical trial, it's incredible. The number of assessments, the number of blood draws, the CSF, the imaging data, we can actually deconstruct this from group means, if we choose to, from group means down to individual responses, and ask, who responded, who didn't respond, what are their biomarkers saying, what aligns, what doesn't align, what are the confounders, and begin to ask those questions. The companies are not motivated to, and I'm an old pharma guy myself, and I understand this, do a full Monty on their data, because there's a lot to analyze, and there's a lot at stake from a corporate perspective. But as researchers, as subjects who are involved, as some of you are, thank you, in these sorts of studies, isn't there an ethical responsibility to get as much information out and ask the questions scientifically that a company might not be getting around to asking, that we've already started talking about in this discussion? So yeah, if I look at the Clarity trial, and I recognize that about 90% of the people, ballpark, are non-Hispanic whites of European descent. Well, let's find out as much as we can about those 90% of the individuals and the response to the drug. And then that becomes part of what we need to elaborate when we diversify in other trials or through registries in primary care, what we're going to learn about people of other ancestries. And if I were to put a wish list, my number one wish, and when we're going to be talking about outcomes of this very important workshop, is how do we as a scientific community compel the making available the biosamples that are being banked, the wealth of data, so that we can begin asking strategic scientific questions so that we really do understand what do the outcomes mean that we're identifying as clinically important, and what might not be clinically important, if we can deconvolute things from group means to smaller groups phenotypically, right down to anchored measures in individuals. And I'll stop there.
Great, thank you, Barry. Great points, and making sure data is available is a theme I think I heard you mention sort of in different ways. And
when I've read some of these papers, I'm actually kind of appalled when I see a statement, and I'm not going to mention which paper this came from, but I know some of you will know who I'm referring to. The data will become available when the FDA approves our drug.
Yeah.
Sorry, guys. I just see that as unethical. I'm sorry.
Yeah. No, data sharing, data availability, very important. The Alzheimer's Association's GAIN platform puts a lot of data out there. I see our CPAD colleagues are on the line as well. Great job of data and making that available as well. So good points. Thank you, Barry. John, are you still? I don't see your hand up anymore, though.
I think John dropped off, but Amy has her hand up.
Okay, he went to a different room, maybe. Okay, Amy, go ahead. The floor is yours.
Thank you so much. I just wanted to kind of piggy off of this idea of data availability. So my background is I'm an assistant professor at Mass General Hospital, and my background is in machine learning analysis. And I just was reading a lot of papers because I also did brain aging studies, molecular studies, and I realized that there might be a need for performing machine learning analysis of available data for AD to develop plasma biomarkers that are outside of, the classic known ones. And so I went through papers that has supplemental data, and I found one from 2007 that looked at 120 plasma proteins indiscriminately. And then I performed machine learning analysis, more modern techniques, to develop model and prediction model and a panel. And based off of my findings, I got funded from, recently from the Alzheimer's Association. Sorry, I'm kind of sick, so my voice is a little shaky, but to validate the biomarkers. But one of the things I was also thinking was if the data were available, we can do a lot of machine learning analysis to develop new panels of biomarkers. So I think that's something that I also would like to advocate for. And also, I also applied for funding at the NIA for a new to field category for AD, and to try to do proteomics more indiscriminately from using banked plasma samples. And at the Mass General Hospital, one of the great things is we have this amazing resource through the biobank where people have plasma, serum, DNA, and some PBMC stored from, not specifically for AD, but from prior, like when they went to the hospital before they had any diagnosis. So you can do these studies where you can look before, well before diagnosis, and to try to develop new biomarkers. And then I spoke to Dr. Roberts at the NIA about my grant proposal, and she gave me the great feedback that one problem with our institution is the lack of diversity, because like we're in Boston and the patient population, and I see it as well, because when I go to the biobank repository from my institution and look at the population, it's very not diverse. So she suggested to like partner with other institutions with more diverse samples and things like that. So I really wanted to echo this idea about, you know, like partnerships, collaborative work with across institutions that can like share resources, but share resources in a way that is more inclusive, and the data availability. So thank you so much for, you know, it was really enlightening for me as well as I think about my future research.
Thank you, Amy.
John, I see in the chat that you're having trouble. If you'd like, you can put your question in the chat since you're having mic issues.
I'm happy to read it here too. So my question is, how can we structure the use of biomarkers to try to detect groups that may be less resilient to beta amyloid toxicity and may explain some of the divergence in the cutoffs?
Chris, maybe Fanny has some comments on this as well, but I'm wondering about developing a profile of biomarkers, say, as well as other factors, environmental, exposome, polygenic risk scores, and putting these all together to try to develop a prediction formula as to who might be responding to some of these therapies, who might not be responding to the therapies. There could be racial, ethnic differences. Polygenic risk scores may capture some of that, but there's a variety of factors that come into play. And I think machine learning approaches can bring all of these together, because it's going to be much more complex than a simple, here is the predictor who's going to tell you what's going to happen to you as an individual going forward. So to address John's question, I think it's going to be much more complex, but I think we're getting there. And, you know, getting to question 2 and 3, that we need to get outside of the universities. We need to get out into the communities. We need to capture real world data on individuals, people who are dealing with and living with a variety of other medical comorbidities that may very well influence the effect of a drug or may influence the performance of a particular biomarker. So I think the expansion of data resources to larger communities, more generalizable communities, both racially, ethnically, and in the general community, I think will be an advance. And we have heard some of that yesterday. So hopefully that's the direction in which we're moving.
Yeah, I really agree with that, Ron. I think we've been simplistic to a fault, because we've wanted to have a few biomarkers and validate them across the board, but we're not seeing what we're now seeing. So now I think expanding and capturing orthogonal or synergistic pathways on a panel, it doesn't need to be many. And I think the approach of machine learning is a really good one. And what we've done is we start with a lot, but then you cut it down, you boil it down to a minimum sufficient set of predictors that sufficiently, to a satisfying degree, can separate your groups. And not perfectly, you know, there's some concession there because you can have overfitting with too many measures. So you get down to smaller number of measures, but then more generalizable to a certain extent. And I think the other thing, Ron, is I consider disease to be really continuous, and that's what we hear from our patients. It's not these buckets. So we also need to potentially think about disease differently. And this comes back to the point that I made yesterday about when are we ready to intervene on asymptomatic or very subtly symptomatic disease. That will shift as to how we quantify pathology and also what outcomes we're basically quantifying. That is very much related, I would say, with the toxicity of the treatments, however, right? So you do need a higher threshold of pathology if the treatments are both expensive, going back to the economy of all this, and toxic. But if we can reduce the toxicity and the cost, then why do we need to intervene on fully fledged disease or impairment? We could do it in very, very early stages, like all the other systemic pathologies that we intervene on.
Great.
Great point. And I think a lot of the machine learning, AI, the modeling efforts and looking at, so I mean, there are new, say, plasma biomarkers emerging almost weekly that we're hearing about, and incorporating those into a model that can help understand a little bit more about disease progression, what's the biology that's happening, again, long before you see any symptoms that manifest themselves, and understanding what all of those mean as they emerge, and having models that incorporate those emerging biomarkers, because we don't have the answer right now as to which one or two is going to predict where you'll be at, but understanding in a more modeling approach to see which ones would be most effective in diverse populations over time is where we really need to be. And there are a lot of efforts that are going on like that.
Chris, can I ask you a question or maybe a request to the association? And this could be a multi entity effort. Going back to the question of disclosure of results to the participants, and I think to the patients, we're at a point where if we want to do things differently and really incorporate the plasma biomarkers, we need to strengthen our partnership. And I think we really ought to put effort into the communication of results to individuals who are not scientists, but intelligent. And if we communicate it in a non-jargony, and I like visuals when it's like, even as a scientist, I like visuals more than words. And so I think if we were able to boil down even the uncertainty of our findings and the variance that we don't fully grasp into nice visuals, it's going to go a long way to building this rapport with individuals who are generously contributing to the research. I like the example of TPA for stroke. We've all done it. Anyone who's done neurology, in a matter of seconds to minutes, you need to convey to an individual their risk and benefit from a treatment, and they need to choose, and you don't have a long time. I think that because dementia unfolds over decades, we're slow at doing some of this. And in some ways, we don't have that much time because maybe the therapeutic window is more narrow than what we would think. And so if we can convey to someone who's never heard of a clot busting medication, the risk and benefits, and have them make an informed consent, I think we can do a better job of conveying how we as a field think about dementia and why these fluid biomarkers could be game changing and what our uncertainties are out of N of 1, right? So there's these figures with like people and showing how many get harmed and how many get benefit in a visual form. So you don't need to know statistics. You can just look at it and understand to a certain extent. So visuals would be really fantastic of some potentially even advanced concepts. We could even incorporate machine learning into this. I mean, that's like more advanced. You know, we could start with more basic, but it would be really wonderful to do this and then get our IRB approvals to allow us to communicate.
Great points, Fanny. And yeah, we actually, so the association is acutely aware of this issue. And coincidentally, one of the efforts that I lead at the association is a work group on clinical meaningfulness, of which Ron had presented some of the data on one of our manuscripts that we had published. And right now we have submitted a document that is focused on how to communicate risks and benefits to patients, for physicians, how to communicate risks and benefits of emerging treatments right now, so that there is some standard language. There's a lot that we cover, and I won't go into it here, but one of the things that I just thought of while you were talking is, it is the time that it takes, especially with Alzheimer's and their patients, and making sure that there is adequate time with the patient and the family to communicate these results in a way that meets them where they're at and whatever level of understanding they have. Because in our conversations with physicians and patients and PCPs, you find that some patients do like to get into the details of, well, what are what are the statistics of my chances of getting this? What does this ApoE mean? And then other people just want a much higher level view, and they don't want all the details. And understanding where your patient and family is at with those discussions, I think, is really important. But having some nomenclature that is sort of acceptable and is positive, and it reflects the full information about what you would want to communicate about risks and benefits, I think we are moving in that direction. But there's so many opportunities out there that we really want to try. We have to start somewhere.
Great.
And I was just looking at, scrolling through, Luke said, reminder, we will break at 12:35 and return at 12:45 for breakout session reports. So I think we're right at about that point. It's probably a good place to just to wrap this up. Thank you, everybody. Thank you for the discussion. And I look forward to seeing everybody now on the other side. We'll go have our breakout reports in just a little bit.
Very good. Thanks a lot, Chris. Okay. Bye-bye.
Right. Thanks.
So basically, my name is Joe Hunter. This is the breakout session for Economics, Regulators, and Payers panel. I'm from the Centers for Medicare and Medicaid Services. Other moderators will introduce themselves as they step up. As a general disclaimer, my comments and questions are intended for brainstorming, are not attributable, and do not represent the views of any government entity necessarily. So please do introduce yourselves to each other in the chat, who you are, where you're from. We want to hear from as many people as possible, particularly those who have not had a chance to voice their input so far. As a reminder, keep dialogue, including in the chat, civil. Of course, we all will. And to start this off, we are actually going to not start with the G&O slide. We're going to start with an update on digital health technologies from Dr. Michelle Campbell from the FDA. So she will give a brief, introduce herself and give a brief on the DHTs. And then we will put up the GO slide and open the discussion. At the very end, we're going to ask to do a round robin almost. Give us some key takeaways as you see to update this gaps and opportunities. We'll start with the speakers, but I want everyone to chime in. And it's really sort of like a lightning round of one bullet each at the very end. So with that, Dr. Michelle Campbell, please go ahead and take it away for a few minutes.
Thank you, Joe. And good morning, everyone. And thank you for staying along for day 3 of this meeting. My name is Michelle Campbell, and I'm the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience, which is in the Office of New Drugs in CEDAR at FDA. And so I want to spend a couple minutes real quick just letting you know what is happening from an FDA perspective on the utility and the growing advancements that we are seeing in the use of digital health technologies in clinical trials and medical product developments. And so first and foremost, I want to say that we do have a digital center of excellence. It is based out of our Center for Devices and Radiologic Health, CDRH. But they do work closely with all 3 of the medical product centers. And so I will be referring for the next few minutes at things at a medical product level. So that is CEDAR, our CBER, our next center is CBER, which is our Center for Biologics, and then CDRH. And so our 3 medical product centers align on a lot of things, including development of DHTs, the development of clinical outcome assessments, of what's needed. And so when it comes to DHTs, we are aligned on the approaches. And so we actually see that DHTs can transform how we are studying investigational clinical trials and medical products. And so it can allow for remote data collection, decentralized trials, improve access to clinical investigations, improve clinical investigations of various endpoints and biomarkers, and help capture real-world data and patient-generated health data. I want to inform you of how we define what digital health technology is at FDA. And it is a system that uses computing platforms, connectivity, software, and sensors for healthcare and related uses. And so this could be used in a medical product. It can be incorporated into a medical product. And that's where our Center for Devices would come into play. It can be used in the development piece. It can be studied in a clinical trial, and it can be used as a companion or adjunct to medical products. And so those are just some examples. It is a large spectrum, DHTs. So it can be taken in any form, from an actual hardware, such as a continuous glucose monitor hardware, something that is put into the skin, to a software, which could be electronic data capture of an ePRO or an eCOA, that software. And so it takes many forms and shapes, and can go down to consumer general wellness products, such as our Fitbits and our Apple Watches, to electrocardiograms, to remote sleep. So there's the term digital health is very broad, and the technology captured on it is very broad. Our DHTs should be fit for purpose when we are using them in clinical trials. And again, fit for purpose is the conclusion that the level of validation associated, and in this example with the DHT, is sufficient to support the proposed use in the trial. And this is really important because we definitely hear the buzzwords digital biomarker a lot, to encapsulate all digital health technologies. And I would encourage us to rethink the utility using that word. And if we are going to use that word, we're using it in its correct nomenclature. So what determines if something is a biomarker or a clinical outcome assessment, which is the 2 big areas we're seeing DHTs emerge right now, is based on something called its context of use. What exactly is it measuring and in what situation? So some digital health technologies are doing more of what a biomarker would do. I think continuous glucose monitoring and the blood glucose values we see is a great example. That's a very solid example of that as a biomarker. But when we're talking about something that is going to support a clinical trial efficacy endpoint, it is now really more on the side of that clinical outcome assessment, and that the DHT is the way we're collecting the data, but we're going to form an endpoint from this data to support efficacy. And so it is really important to make sure that there's an understanding of what you are measuring, and to help determine which path. And with that, for DHTs, and I'm going to wrap this up, that are going to be used to support clinical trial endpoints. Our conversation that we've been having for the last 3 days on clinical meaningfulness is extremely important at this time. We need to make sure that what we are creating with this clinical trial endpoint data that is being captured from a DHT meets what our definition of clinical benefits, so an improvement in how a patient feels, functions, or survives. And we also want to make sure that that is clinically relevant or clinically meaningful to them. And there are ways that this is being assessed, and I will say that we are collectively working together on multiple fronts, the agency is, in the pre-competitive space, on how do we really take, for the efficacy side, to take the DHT information, how do we know what data is the best data to use, what is that context of use, and how do we form that endpoint? And then from there, how do we interpret it? And so we need to make sure that we leverage some of our measurements, good measurement practices in more traditional COA landscapes a little bit when we're looking at our DHT data that's going to support clinical trial endpoints. So that is a very, very quick synopsis on DHTs and its utility. I do think that DHTs has a great opportunity in the neuroscience space. Obviously, there's some areas where we naturally are seeing them, but they do open a way to collect valuable information and really capturing that patient and person's lived experience of what their capabilities are. But a lot of times what we know is the detail and level of evidence and validation work is what is really going to be what needs to be well thought out in determining which path we're taking in this informative digitally collected data. So Joe, I turn it back to you. That was a very, very quick synopsis of DHTs.
Thank you. That was clear and informative. I'm going to take the moderator's privilege of asking a quick question. So as I understand it, the legitimate outcomes that you've had across FDA in terms of, are you improving how a patient feels, functions, or survives? It seems like this really doesn't change the outcomes or endpoints of trials or standards, right? By how we judge whether a trial succeeds, and if so, for whom. It's really just an additional way to produce those outcomes. And then, but it's a similar evaluation. Is that true, or is there more nuance there that I've missed?
I think there's some minor nuance that I think is not, I don't think today is the day to get into some nuances. I would say that DHTs offer an opportunity to perhaps capture something that we would not have been able to capture with maybe more of our traditional clinical outcome assessments. We've heard about, they bring in greater sensitivity to what they can collect. But in the end, when it comes to supporting an efficacy endpoint, our what we have to look at is the improvement of patient feels, functions, survives. So it is really, how is this data that is being collected from the efficacy standpoint going to, again, capture that and make it interpretation wise. Again, and then if we're going under the biomarker, what type of biomarker is it going to be? Is it going to be a monitoring biomarker? Is this a progression-based one? Is PK/PD? So again, we still have to think about that context of use and what we think the data is going to be informing and can support. But it is a ever rapidly evolving space. And I really do say that when we do talk about trying to, we're working together in areas, we do have a guidance. I'll put a link in this chat. We have a final guidance on considerations for using digital health technologies, and it talks about the validation and analytical validation work that needs to be done. And then CEDAR has a DHT framework, which really does align to the guidance. And we have our own working groups that are looking at this for the utility for drug development in many ways. So I think DHTs can offer a lot of opportunities, but we still need to make sure that we are minimizing patient burden. We are still collecting informative information. And I don't know if it will solve all of the questions we have, but it may be another tool in our toolbox in collecting informative information.
Thank you very much. And why don't, before we switch to the general gaps and opportunities slide, can we just get some comments, questions from everybody in the breakout session on this topic of DHTs, digital health technologies, and then we can move on to some of the more general gaps and opportunities. So the floor is open for questions and comments. And just go ahead and raise your hand. I think I can see everybody on one screen. And I know also too, not to put any pressure, we do have a couple experts from NIH who know something about this topic. Julie, why don't you go ahead and ask the first question or first comment?
Thank you, Michelle. That was very informative. I wanted to know if you could provide an example of a digital health technology that was used to measure changes in severity of a disease, if there's from other disease states that might be applicable for us to think about here.
So an example that I'll use, there's a lot of work that's being used in Parkinson's right now with digital health technologies. And in the Parkinson's space, there is a shift to go earlier into disease. And with that, traditional clinical outcome assessments most likely are not going to be the most appropriate way to capture clinical benefits. It will most likely need to be shifting more into biomarkers. And this is a very also evolving space. But there's been some work in the pre-competitive space actually that has been looking at because there's really not a definition of what is early Parkinson's disease. There's been some work in the pre-competitive space with industry and academia that has been testing various digital health technologies longitudinally. They're about to enter into their third year of patients who were more recently diagnosed with PD, and to kind of track what that progression looks like, to get a better idea to maybe identify either things that we can potentially measure, support an endpoint, and identify gaps of what we're realizing maybe. And what is really driving some of this that we're learning more about is the non-motor symptoms. And so there are actually what was being found, and this data is helping to support this, is that there are non-motor symptoms that actually emerge first for someone that may not realize that that is going to lead them down this Parkinson's path, but it is the beginning of that journey for them. And we are also in that effort, they're also collecting some normative data to make sure to counterbalance what could be normal aging declines that could occur as well. And so to really balance what is happening in that in the actual patient population. So that is one example that we're seeing where it's not the truly measuring progression over time, but it is emerging, going early, and what does that look like, so we could continue to monitor. I know I'm trying to think of some other things that I'm aware of, but that's just one example in neurology that I'm actively known. Obviously, with Parkinson being a heavy motor movement disorder, that space has been using DHTs for decades, more along with the motor symptoms, so actigraphy, accelerometer, things of that nature. And some of this work is trying to employ it again into these more non-motor symptoms as we start to see development earlier, pre-motor symptom developments into the disease, where hopefully we could delay progression.
Okay, let's move. Any other questions on or comments on this area of digital health technologies? Okay, why don't we can always get back. Okay, Suzanne, go ahead, please.
Hi, thank you, Michelle. I was wondering if you could share what FDA's perspective might be on bringing together multiple features from a digital health technology, for example, a COA, versus a single feature, bringing together multiple features, perhaps more comprehensively assessing a concept versus the preference of perhaps a single feature for scoring.
Yeah, it's a great question. It kind of goes into, if we were talking a traditional clinical outcome assessment, you're suggesting the composite world, right? Pulling different things. And so actually the Parkinson's example I gave, they are kind of doing that. Now they haven't gotten to the point where they've created how to pull this data to form an endpoint per se, but they are capturing different distinct concepts, which is what I think you're getting at, right? So in the COA world, we will be capturing different things, right? Concepts, and trying to bring it together. I would say that, we would want to talk to someone early if that is an approach they want to take, because there's a lot of nuance to that, not just as necessarily in technology, but what is needed from a statistical perspective of that endpoint. And so I would say, come have those conversations early with us so we can, one, bring our internal experts to the table in both the health technology and DHTs, as well as the statistical side that would be needed. And probably begin those well thought out conversations of what it could look like.
Okay.
Thank you.
Thank you also too for the next speakers. When you make a comment or ask a question, if you could just very briefly say where you're from, who you are, where you're from. Any other speakers on this, or should we go to the general GNO slide? Okay, Sue, go ahead. Thank you.
Hi, sure. I'm Sue Peshin with the Alliance for Aging Research. And this is sort of a question both for Dr. Hutter and Michelle, which is, we worked, I guess it was a couple of years ago, on the CGM issue in terms of durable medical equipment and whether folks were using like the transistor piece of the CGM versus an app. And I just sort of was wondering where things are at in terms of evolution in coverage, because, you know, there's increasing use of medical apps that the FDA is looking at. Michelle, it'd be great to hear where FDA is on digital apps lately. And then the transition to sort of recognizing, you know, coverage, whether it's, you know, has to do with the definitions at CMS, just where things are at would be great to hear.
Michelle, I don't know if you have any
Yeah,
on
I
your
can
side to that.
I can quickly start. So, you know, I think we're continuing to learn, Sue, about how we can use app-based stuff. Now, I think in the my last call was literally with Electronic COA Consortium, who are the vendors who develop those apps. So I can say that in a more traditional space of COA collection, and because that actually would fall under the software aspect, I think we do have some familiarity of how that's being collected, because a lot of those that data that's being collected is typically off of some type of provisional device that may be in an app form that a person clicks and they're collecting that. So I think we have some of that information. I think what you're describing is definitely, and as a child of parents who both use continuous glucose monitoring, it is always on their Dexcom app to see what their values are. I think you're really getting at how is all of the technology, now that some technology vendors are shifting to this app-based aspect being used. So I probably don't have the best answer for you. I would actually want to go and talk to some of my device colleagues for a better answer on that. But I would say that, you know, as if it is part of that medical product that's coming in, it will be reviewed, right? And so that would be part of that effort as well. Just as we would evaluate packaging of a drug label. And I see Nada's put some information on as well. So I don't have the best answer for you, but I think it's a question I can always take that back and answer and try to get an answer.
Mm-hmm. And for me too, I think that's a good question. It really is. And it's the heart of some of the things we're looking at now. I don't have a great answer for you, in part because this is new. And generally, we love to have NIH, FDA go first. We don't jump out in front of their army of scientists who are working more closely and diligently with the industry and people in academia on this. And so, and it does cut across different centers. You had to put this in the context of DME, durable medical equipment. Our DME expert, Susan Miller, who I think you probably know, isn't on the call. I would want to run this by her. But I will make some general comments that are known by Sue and some other people, but not by most people generally. And that's that when CMS pays for items or services, it usually doesn't go through a national coverage determination, which my office does. You can just put in a code and it flies through, or it doesn't. Literally, that's done all the time. B, you could just go to local contractors and sort of build from there. And it may or may not even get on our radar at the mothership, in Baltimore right away. And that's not going around the system, it's part of the system. We tend to look at devices mostly. It's rare for my office coverage and analysis group to get involved in drugs, although we have in the past. And there's a whole other center that deals with drugs. So there's many different avenues to go. And when there's sort of a new technology, it's important that we coordinate. And there is someone who's now an internal navigator for things like AI and software as a medical device. But I think it's premature for me to say, here's what CMS would say now about this emerging topic. That said, Sue, I know you know how to contact me and it is a great question. So I'm really not trying to put you at bay, but rather to say, I don't yet have a great answer either. Definitely listening, interested in all of your ideas in how to shape this so that it better serves patients. Any other questions on here on digital health technologies in general? Why don't we put up the GNO slide? It's the gaps and opportunities slide for the econ regulator payer group. There we go. And part of what we were doing now, we don't need to focus on this. We can change this. So just in terms of what are the big takeaway messages that you have learned, you have yet to share for payers, regulators, health economics in general, please go. Any questions or comments in general? Anything to add to these bullets or to tweak these bullets? And I'm going to, if people are being shy, I'm going to call on my fellow speakers to see if they have, these bullets came from them. We had 3 speakers, we've got 3 bullets up here, and I think they're interesting. But if there's anyone who wanted to sort of, hey, we could add a 4th one, we could change this one, that type of thing, please do. Sue, go ahead, please.
Thank you. When I saw number 2 yesterday, I was a little I was a little struck by it. I understand that the national plan on Alzheimer's disease talks about the costs related to Alzheimer's, but the framing of this seems just kind of particularly targeted. And in that, you know, because we're talking about sort of Alzheimer's disease, and given that this new drug class was placed under a coverage determination and everything, I wasn't sure if this was, so the context of this meeting was Alzheimer's disease, right? So it's not really talking about all types of treatments for Medicare. And I'm just wondering, like, do we do this in cancer and heart disease and all these other different areas? Why are we, you know, we have an aging of the population. It would seem like we need to figure out how to meet the demand of that rather than talking about how do we constrain, you know, and especially in terms of NIH's remit, it just sort of struck me that the way that this was framed seems to present Alzheimer's patients as a math problem to be solved.
Mm-hmm. Well, thank you. The person who actually created that bullet is, I just looked, is not on the call today, so not sure how he would answer that. And I know it references Medicare. I am from CMS, but that's not coming from me. So thank you for your input. And I'll just reiterate, though, that you know, in our office, there are parts of CMS that does look at cost. The Innovation Center has a different authority and explicitly can look at these things. They want to improve quality and decrease costs ideally, but you could maintain quality and decrease costs, or you could just improve quality. So they have more authority and things to do stuff like that. In our office, we literally, someone once said there was this long report by ICER up in you know, Harvard Mass General Hospital, and someone, as a stakeholder on the call, was like, "Well, you guys are just following ICER," and I'm like, "Guys, I only read about half of that," and they were stunned. And I'm like, "Yeah, no, there's half of it that had nothing to do with what we do at all, and I didn't even read it." So, so thank you for those comments, Sue. I think that's a well articulated point that is shared by others too. Julie, go ahead, please.
I just wanted to follow up on Sue's comment. It sounded like that, and Sue, please correct me if I'm wrong, that she wanted to bring in perhaps a different aspect of these innovative treatments that are coming into market, which is that we don't quite have a good grasp on demand across the population, what aspects of the drugs are valued the most, what matters, which came up a lot in the session this morning. And Sue, I'm sorry if I'm kind of changing what you were saying, but it does potentially relate to this, different than how we afford it, but about, what is the demand across different groups? How do we ensure access? What aspects of the drug are most valuable? Excuse me.
Okay, thank you. Perhaps going to the third one there, development of measures and models that quantify the short and long-term meaningful health and financial benefits of treatments, which could be used by various stakeholders. Any further elaboration or addendum to that, or any comments along this theme?
I guess, Joe, we've done a lot of work on patient preferences in Alzheimer's. Jeff Cummings talked about the work that we did yesterday, citing one of our papers. Michelle's been involved in that. And it's not 100% clear that what we need is more measures, as much as we need to have maybe a core outcome set for the measures we've got, or to understand that it's a heterogeneous disease and that patients care about a lot of different things. We have to be open to different possible endpoints. The models, that's more of a Julie thing. I don't fully understand models in this sense, but I think that we should also make ourselves aware of all the work that has been done and is ongoing, and understanding what matters to patients in a very rigorous, quantitative, methodologically sound kind of a way.
Okay, Michelle, go ahead.
Yeah, so this is a unique opportunity for me because my actual background degree is in health services research. So I slightly still remember my learnings of health economics and modeling. So I'm going to try to pull my thoughts together, of blending to kind of Russ's point as well, is that, because my first question is, well, not only do we need new measures, but where are the gaps? And that may still be kind of evolving as we begin to see approvals of new therapeutics, right? Because we're not fully sure what all of them are going to be. I think we all have an idea of what they are and that we know, but I think we're just at the beginning of some of this. So my thoughts kind of blend 1 and 2, which is, how do we really make sure we are capturing concepts that will help all stakeholders and that are standardized enough that work for FDA, they work for clinical practice, they're ultimately going to help inform payers, including CMS, in coverage decisions. It's going to so that's how my mindset is kind of thinking about this, is that folks sometimes think that developing new measures is the answer, and it may not actually be the answer. There may actually be some existing things that can be pulled together and leveraged, and perhaps it's actually what needs to be done is better standardization or approach of how to utilize some of these measures to really be informative. My clarifying question would be to Julie, or to those who need the data to feed those models that are informative, looking at, how this does improve long-term care cost benefits, or overall, are we delaying, I'll give an example, like, are we delaying nursing home admissions because of intervention of medical product, right? So what is missing, or what would be good that we would want to think about at the earlier stage to start building that data for those future models?
Okay, all right. Thank you. Yeah, it seems when it comes down to the clinical outcomes, I mean, we're very concerned about that also. There was sort of a call to have a core outcome set that could be used across trials, which is easier to compare across trials. There's always some caution when you're trying to compare. Oh, here's like this drug, I'm just making these up, got a 25% improvement in this randomized group using this outcome measure. Here's another trial with, heterogeneous patient population, different randomized group, and you've got an effect size difference there, say like 35%, and it may be a similar outcome or a different outcome. How do you compare to that? You really can't very well with a lot of fidelity, right? And one of the things that we just heard from this morning was that they want to see head-to-head drug competition there. So a core outcome set would help in cutting across, comparing trials a little better than we do now. Ideally, it would be something that is more sensitive, and maybe digital health technologies will help with that in the future. And it will apply to all or most subgroups. That was mentioned yesterday too. That may be a hard standard to get for an outcome measure. Yes, it applies to all subgroups and to, and so it could be that you've got a core outcome set of some basic things. You may have something different for subgroups. I don't know. And then you've got something where we are moving to earlier stages of disease. If you look at the FDA and also sort of the Alzheimer's Association, NIA new guidelines in the staging system and stuff, it seems like we're moving in general in trials that we're targeting stages 2 to 4, with 4 being mild dementia. Michelle, correct me if I just messed that up, to stages 1 to 3. So the whole focus is moving one step earlier, where 1 is asymptomatic, right? And then 3 may be MCI, and 4 is mild dementia. If I got the numbers wrong, Michelle, just tell me. So we're moving earlier, and then if you're moving earlier, then having that outcome of, when do you convert from, one point to another, gets a little more difficult. You may have entire trials where very few people progress to actual mild dimensional, where you have functional deficits. And yet you may see something that, wow, that is a substantial change within MCI itself. Or you may have some cognitively unimpaired people who do transition to MCI, and there's a transition, the type of thing Ron Peterson was talking about yesterday. So I think it's a tall order. I for one have more questions than answers on that, but I wonder if anyone else wants to weigh in on those things. Alicia, did I pronounce your name correctly?
Hi, I'm Licia.
Thank you.
Licia Neumann.
Okay.
I don't know all of you, but I joined the Alzheimer's Association last year, and I work with health services research. So thanks for bringing that up, Michelle. Some of my notes here are actually very aligned with what you were saying. I feel like one of the important things that this meeting has done is to bring the different perspectives that need to be considered as we discuss outcomes. And they are so different, and which of course can lead to conflict, but I think we all have something in common here, no matter if we are patients, caregivers, advocacy and support organizations, or payers and regulators. I think we all want to see that individuals with dementia or MCI have timely access and equitable access to quality care. And I think the important point of all these discussions have been to understand what really matters to whom and when. So when I look at the bullet number 3 here, I think before we discuss measures and models, I think we need to have a good agreement on what are the outcomes we are actually looking for. And then what is the metrics that we need to assess to what extent we are actually achieving those outcomes with everyone. And then I agree with what has been said, that not necessarily we need new data. I keep wondering, do we need new data, or do we need to work together to better use the data that we have? And how can we better integrate data in a meaningful way? Because models will be as good as the data that we input on that. And so if we are missing the point on what we really want to evaluate, then there is no statistical model that is going to give us the information we need. Yeah. And then finally, I just always like to say that from a health services perspective, health services research perspective, we need to think on what are the decisions we are trying to inform. And remember that decisions are being made by patients, physicians, payers, and government anyway. So how can we ensure we are actually bringing the data and evidence that they need to make the best decision, considering, of course, how diverse this environment is, which I think was another important point of this workshop.
All right. Thank you. Russ, and then Michelle.
just quickly, one little thing and one bigger thing. So little thing, Joe, you said something that I've been saying a lot, which is that we're going earlier. And if you say that in front of a population of dementia patients, some of them are going to get mad at you because it sounds like we're leaving people with more advanced disease behind.
Yeah.
And it's certainly the case that there are drugs in development for people with moderate to severe disease, or at least the symptoms thereof. And it is also the case that they feel like the DMTs at least are leaving them behind. I think we just need to be, like, it's a technical point, but we should be careful how we talk about it.
I actually think that's really good as maybe a bullet to say, yes, there's this focus. There were 27 failed trials for a combination of reasons. One of those reasons likely was we're getting people at moderate dementia and these things targeted just didn't work.
Yeah.
Now but you're right. It's like, as we move earlier, maybe there's other things that can be targeted, maybe in combination that can act. So don't forget the patients with moderate disease.
And the other point is, and don't forget the people with either atypical presentations of Alzheimer's or the conference, I just looked at the title, it's AD and ADRD.
Yes.
So there's other dementias out there that have different presentations. So the work that we're doing on patient preferences and the core outcome set are all about literally Alzheimer's disease, pure, and some people have to have diagnosed Alzheimer's disease. But there's a lot of related dementia out there that we don't want to forget about those folks either.
Yeah, no, that's a critical point too. And in fact, in some of the studies where, large registry-based studies we're seeing, people are saying, look, there are subgroups there who would have been excluded from these trials, these pivotal trials for good reason, perhaps. And they don't quite fit this outcome or that threshold. They present differently. So the clinical diagnosis really has to be tailored to them. And there is flexibility to include those because, yes, do that research, collect that data while you're treating those patients. Michelle, please go.
Yes, thank you. And I would agree as well, Russ. I was recently at a different disease meeting a couple weeks ago in the same scenario where they have a population it was in Duchenne muscular dystrophy, and they now are having adult patients, and they say, hey, don't forget us, right? We're still here. We still have concerns, that development. And I think it's a very fair point in approaching how we really think about this. And so I think that also suggests, to the point, the challenges that Joe was pointing out is that some of the things that we were trying to convey in the new early AD guidance draft is that we unfortunately may have to approach how we measure things slightly different depending on where you are. And on top of that, and Russ has heard me talk about this a lot, is those needs of that person and their families is going to shift during the progression of the disease, right? And what their needs are and what actually they would view as a treatment benefit, right? What would they be willing to do? That is going to look very different over that. And I think we hear that in our progressive diseases, right? That looks a little different, and we have to understand. I also think this conversation really also suggests to make sure that we are sharing data, that we're data sharing, choose your preference where you want that to go, but that we do need to make sure we're leveraging existing data and pulling it and looking at it, because that can help us identify the gaps to the models that Julie would like to develop. And so she needs data to do that. I do think it also helps us be able to improve data collection quality, so we can, as the point that was made, we can only trust the data that goes into the model. We need to make sure we're collecting good data. So I do think there could be some existing things that are already occurring that we can think about to help answer some of these questions. But I do think that we could run into some, I like core set ideas. I think how we practically implement something like this in a progressive disease could raise some challenges, because measures are going to shift over time of what is really capturing. And I think that was what things we were thinking about, because really sensitive measures early on are probably really good, but they may vary over time. But if we can at least agree on capturing key critical other maybe data that could benefit in the long run, that can always be informative as well.
Okay, just
Hi,
a
Joe,
reminder.
I want to mark.
And Jenna, I'm going to let you on there. Sorry, I didn't know who was just speaking. Just want to remind people we're going to actually return at 12:35,
4
we end the
minutes, yeah.
session. And at 12:45, we'll be going back to the general group. So there'll be a 10-minute break starting at 12:35. It is 12:30, 12:31 right now. And Jana, please go.
Can you
Yeah,
do a
thank you very
lightning
much.
round, Joe?
Oh,
Sure.
yeah.
Do your lightning round.
I'm going to be quick, Marcel. So really quick, I'm a health economist. I work for AbbVie. Alzheimer's is very close to what I do. And, you know, I just wanted to have comments from the group here, from the speakers about just having this outcome measures be part of trial, because we know that these are very short-term trials, and they, you know, we're looking at a disease that is progressive in nature, and as mentioned, it's changing over time. So the outcomes reported could be changing. So what is measured from a trial perspective, and then supporting evidence by using real world evidence, you know, how important that is, because that also feeds into the models. When we talk about long-term evidence, that's clearly important because we do not know the impact of these treatments over time. And then the second comment I wanted to make is regarding the spillover effects, which is there is a huge care component here. And traditionally, most trials are not looking at that, and how important that would be to be, you know, included as a perspective, and also because the carer perspective changes over time on what is demanded. So as a society, or as from CMS perspective, and we're thinking about this, there are huge implications, not just from the patient perspective, but also from the carers who are taking care of these patients.
Okay, lightning round. So one quick bullet. We're talking like 15 seconds each person. Russ, I got a couple from you, but you can pick out your favorite from what you've already said.
Favorite bullet is that people ought to brief themselves on the work that already exists and make sure they're aware of the work that's already in progress before suggesting new things need to be done.
Okay. Next. I can call on people, but I don't want to do that. But just go ahead. If you've got something to say that you haven't said already, or even if you did. And if there's anyone who hasn't spoken yet and wants to put in one thing, support something that's already said, or bring up a new thing, now's the time to do it.
Come on, folks, you can't let me be alone.
That's right. We've got some good things here. And what I'm going to ask, Marcel, I don't know if you have anything that you want to add, but what I'm going to ask is if the NIH staff and moderators could stay on this line at 12:35, 1 minute from now. We're going to just, I've got some bullets here. You've got some notes. What are we going to say then for a little 5 to 10 minute pitch in front of the whole group of here's some ideas, some bullets that people suggested we focus on. So with that, I will let everyone go.
So the breakouts automatically close. We can set up another room for you if you want us to.
Let's do that then. Let's just have very quick, can we summarize some bullets here and coordinate how we're going to present this at 12:45. For the rest of you, we'll see you in the group thing at 12:45. Thank you for all of your input here.
So each presenter, as a reminder, try to keep your remarks to 10 minutes. Really just give us the high level major takeaways that came from the group, especially emphasizing gaps and opportunities that may have been missed from the initial draft slides to what you have now. And after those 5 summaries, we'll open up to general discussion before closing out the meetings. And at the close, I'll talk about some next steps. Rosie?
Terrific. So this is the gap and opportunity slide that was developed during or right before session 1, and that together during the breakout session, and thanks to also the participation of our lived experience panel, we were able to expand upon. The first point is cognition, equity specifically in cognitive measurement. And the problem or the gap is that there is a need for skillful and informed adaptation and cross-cultural validation of cognitive tests that measure clinically salient outcomes, and that there's a lack of harmonized longitudinal assessments for diverse Americans. And some of the initial opportunities were discussions about developing novel cognitive testing approaches and adopting novel cognitive testing approaches and technologies, particularly those that have shown to be sensitive and specific enough to detect meaningful cognitive change that is, and to track it over time that would be consistent or with disease progression. Also to facilitate collaborative research amongst investigators, particularly across disciplines, that study how AD and ADRD present and progress in diverse Americans, and recognition that this could really optimize the cognitive measures and accelerate the availability of these measures, particularly those that have shown to, good validity data and reliability data. We expanded upon this thanks again to the expertise of our lived experience panel, that they said, you know, we really should be able to provide results moving forward to those who want it, so that they can integrate their results into their current life and into their future planning, etc. They want to know what went up and what went down, and what does that mean, and what we are tracking and why we are tracking it. And perhaps even something that they can take home with them, as that they can, you know, process later, for example, or process with their loved ones. So developing ways to return results to research participants. The point was also made that we would develop a continuous improvement loop in our research programs to learn about what aspects of the research can be improved by persons that might have different perspectives. Even talks, for example, about ratings and for different parts of different study procedures or different cognitive tests, etc. The second gap that we discussed was the need to improve upon precision of repeated cognitive measurements by reducing practice effects or capitalizing on practice effects that are produced by many cognitive assessments, and the notion that this could reduce time and burden. Dr. Gibbons in his talk mentioned or taught us about large cognitive item banks that can be adaptively administered and tailored to a person's level of cognitive ability with precision, and that this can be repeatedly administered without practice effects or with reduced practice effects, and that this could enable high frequency longitudinal assessments. It was brought up during our time together that adaptive testing can tailor items and give tasks to people that are more personalized in nature, that are better calibrated to the current ability of the participant, recognizing that one size doesn't fit all with cognitive testing, and that research participants might have a hard time with some tests that might be too challenging and could experience negative emotions such as humiliation when they notice that they can no longer do things that they were once able to do. And so the adaptive testing practice can help with that. Other statistical techniques such as differential item functioning were brought up, and that this can take items and test them across languages and cultures and subcultures, and you really evaluate whether there are items that might be biased and not necessarily differentiate people with high versus low abilities. And this has been done, and Dr. Gibbons described how this has been done in other, for example, in mental health. It was brought up that in cognitive assessment specifically, that could be a little more tricky to use, such as because memory, for example, is a very necessary construct to measure, but might be more difficult to do using this method. And concerns that, you know, if we shrink batteries down too much, we might dilute information. But there was an indication that data has emerged about promising correlations in using this method and traditionally administered neuropsychological tests. And so really keeping ourselves as informed as possible. And again, those multidisciplinary and interdisciplinary collaborations that can help us expand our own windows of reference and grow in development of cognitive testing or the use of existing cognitive measures. It was also brought up that practice effects might be a valuable cognitive construct, particularly innovative ways of measuring diminished practice effects might be particularly useful. And so reminding everybody about the literature of practice effects and some of the utility that has been shown by certain groups in measuring practice effects in more innovative ways as of very recently. The third point, the third gap we didn't get to talk about, so sorry about that, but I do invite Dr. Germain to make a point, and it was about measuring cognitive change dynamics using digital tools. And so the gap is that there is a need for novel and clinically meaningful outcome measures that combine cognitive function with health and physiological factors, including the development of tools that are more naturalistic and ecologically relevant. And Dr. Germain makes the point that there are scalable digital measurement tools that might allow us to move beyond traditional single time point assessment into a more content-rich and dynamic model of behavior that more closely matches clinical phenomenon and everyday cognitive functioning. Unless Dr. Germain wants to add anything to that, I'll stop there.
I might add one point just to connect it to the lived experience thing, since we didn't get to discuss it. But I think the other thing that's nice about a cognitive change perspective is that you can now integrate the everyday experiences of patients and participants. You know, so it's not just that my cognition is stable all the time and I'm getting slowly worse, but there are good days, there are bad days, there are stressors that might make people's cognition temporarily a bit poorer. And being able to integrate those everyday experiences, I think, is another way to integrate the participant perspective. So hopefully that is something we can work as an opportunity as well.
Great. And I know other people had discussed the role of cognitive assessment in the context of trials. For example, the Payers, Regulators, and Health Economics group. Michelle was discussing the FDA perspective on digital health technologies and advancing digital endpoints. So I'm sure we'll hear a little bit more from them. And again, focusing on outcome measures and how patients feel, function, and survive. So we'll talk a little bit more in that group about this as well. Also something, Laura, because you're on, I'm going to say this because I've heard it from you before. We talked a lot about validation and the psychometric properties, and validation isn't one thing. Validation for what purpose and what use. And that's something else I maybe will come back to in the general discussion, but thank you. Our next session, Elena Benford, I don't know which one of you is presenting, but the everyday function session.
Great, thanks, Luke. If you can go ahead and put the gaps and opportunities slide up, that would be wonderful for the everyday function group. Okay, well, maybe I'll go in slightly different order. Instead of starting with the gaps and opportunities, I will begin with, there it is, wonderful, thank you. So, but prior to our discussion, these were the gaps and opportunities that were introduced throughout the meeting. The first is really talking about the development of approaches for multi-dimensional assessment of everyday function, inclusive of emotional, psychological, and social functioning that incorporates domains that are prioritized by the person who is living with dementia, cognitive impairment, etc., including the maintenance of self-belonging and engagement in meaningful activities. And this one was really posed as more of a coming from the researcher angle, if you will. The second one is put forward as more of an opportunity to engage in an ongoing way with the community, to ask folks who are having the lived experience of dementia or perhaps starting into a trial about the process and measurement of everyday function. So in that case, the emphasis would be on refining and applying approaches to ensure that outcomes measured are meaningful to, in fact, the folks who are either living with dementia, cognitive impairment, or maybe the precursors to that experience. So that's where we started. In terms of our discussion, which is where I'm going to spend most of my time, we had a wonderful group of experts on our call, many of whom have been working in the field of measurement and everyday function for quite some time. There are 4 topics that I wanted to just put out there, and then I'll repeat some of them with a little bit more detail in a moment. The first is we had a lot of discussion about how audience matters. So if you're thinking about everyday function and measurement related to everyday function, you could be thinking about the audience, of course, and primarily of those who are living with dementia, maybe their family members, maybe folks who are considering participating in a trial. And what you would want to measure from their perspective might be different, for example, than the measures that would be of most interest to regulators or payers or others. So we talked about the fact that the audience for these measures really matters. We also talked about how we may in fact vary in terms of how we conceptualize and measure and even think about everyday function. So Dr. Riley in her presentation on day one sort of expanded our thinking for many of us about how we conceptualize everyday function, including areas of really great importance, including the social aspects of life, as well as how we communicate with others that might go beyond a more traditional examination or measurement of everyday function that tends to focus on things like activities of daily living or instrumental activities of daily living. So how we define everyday function matters, of course. And we also talked about whether or not it might be important, and I think the group agreed, to come up with maybe some common measures across studies, but of course there's a lot of challenge to that, and I'll say a little bit more about that. And my last sort of overarching point is about the challenge of sustaining work in this area. A number of people mentioned how funding, consistent funding and significant funding would be valuable in terms of the development and the ongoing expansion of measures that focus across everyday function and are really informative to the clinical trials that we have going on now and in the future. So I'm going to walk us through some of the more detailed comments and discussion that we had. One is that we, at the outset, and this will not surprise you based on the meeting over the past few days, at the outset, we acknowledge that, of course, everyday function is very important, but is often not actually included in many clinical trials. We're talking about drug trials, but also non-pharmacological trials. And so we'd like to see more of the incorporation of these measures into the design of trials from the get-go, for example. And as I mentioned before, there really is a gap between the stakeholders in terms of participants in the study and maybe those who are running the study or regulators in terms of what they're looking for in terms of everyday function. And that goes back to, again, to that point of who is the information being generated for, right? So if you are looking to measure everyday function, who is the ultimate audience, and for what purpose are you trying to gather this information? We talked a fair amount about how measures we hope could be sensitive in many ways, including over time and across disease stage. So we note that perhaps there might be some significant gaps in terms of what we need to know, for example, at the early stages of dementia, what measures are there and available to help us understand maybe some of those critical changes that are happening with everyday function that might be informative to the person who is new to experiencing such symptoms and could be informative to a research body more broadly. We also acknowledge that priorities can change throughout the course of illness. So from the lived experience of living with dementia or cognitive impairment, somebody's priorities at one point and what they want to measure and what they want to focus on in terms of function may change and be quite different later on. So I think a couple of our speakers, as well as folks in our breakout group, talked about this, that what somebody cares about maybe 6 months after diagnosis versus perhaps many years into illness may vary. And what does that mean for how we collect that information over time? And I would also just note that when we talk about common measures across these different audiences, we hope that having maybe something like a set of measures that could be in common across many studies, what would that look like? That could be beneficial across different audiences, including Medicare, including those running clinical trials, perhaps FDA, maybe community-based organizations, in addition to folks who are living with dementia and their families. And you know, if we had this sort of minimum set of measures, could we then couple that with maybe another set of measures that might be really catered or tailored to the individual experience of folks who are participating in trials, et cetera. So there was some discussion about what types of work we should do in tandem with each other, perhaps. And, you know, we know that not everybody sees measurement development or measurement itself as maybe the most you know, important topic, but we really would like to encourage the research community to pay good attention to what measures matter to those who are living with dementia, and as well as incorporating really sound measurement into their trials, so that we can get the best information possible. And really pay attention to those things that matter to folks across, as I said earlier, the disease progression. And maybe we have to broaden some of our definitions to be more inclusive of the lived experience of those who have dementia. And finally, somebody at the end mentioned that often in terms of measurement, individuals are interested in the thing that they most recently lost and the thing or capacity that they think they may lose in the near term. And so how do we sort of honor that experience for people, whether they're participating in a clinical trial or we're talking to them more broadly? So a great big thanks to our really wonderful group and all the presenters who helped us to set up the gaps and opportunities and identify future directions during our discussion today. Thanks.
Thank you, Elena and Everyday Function Group. Up next will be the Biomarkers Group. Chris, are you presenting for that group?
Yeah, thanks, Luke. I'm going to start off. I'll give you a summary of our discussion, which sort of hops around a little bit on our slides, so we're not kind of going in order. But I thought our and then I'll throw it to Amber and Christina for anything that I missed that we also want to share with you guys. You know, great discussion with this group. There's so much that, you know, when we bring up biomarkers, clinical meaningfulness, there's any number of directions that we could take this, because there is a lot to discuss here. And one of the first, I think, topics that we brought up was related to disease staging and our understanding of the biology of the disease. And amyloid certainly gets a lot of attention, but should we be looking earlier? Which, yes, I think we do. And what other biomarkers of disease pathology are informed that can help inform what's driving the disease? And we understand that there's a value of associations of biomarkers and the disease pathology, but also we need to use that information to understand what's driving the disease, what's driving the progression, which will help then inform ways to treat and stop it. I think longitudinal biomarker data from a variety of populations, diverse cohorts, I think that's what's going to help us better sort this out. So I think that longitudinal data was a theme that I think came up often in our discussion. We also heard some really great comments from our participants in research. Couple that some that were also on our session earlier today, and just the importance of giving back to the community and bridging the gaps in relationships between the community and researchers, so we can better connect with those communities that we desperately need to have more understanding of with our biomarkers, and then also sharing the results with participants, regardless of what community they're in, how to provide results to patients and particularly biomarkers. I thought a great comment made was, I want to know my biomarker results. I want to make my own decisions. I want to use that information, and having that information communicated to them in a way that is understandable, is not biased. I think that's what our patient population and their families really are craving right now. So again, having more longitudinal data on these biomarker results, their performance, and understanding the role and performance of emerging biomarkers and how they perform. we also talked a little bit about understanding the need as a scientific community, being able to have biosamples, biospecimens available to continue to study. That's something too that I think that's a logistical issue that has many facets to it, that if we're going to study these biomarkers long term, those samples need to be collected and they need to be analyzed and results given to the community in a way that everybody wins and benefits from this, not just a select few that are, say, developing the biomarker. So I think we heard that loud and clear as well. So, and then to a little discussion on data availability of biomarkers, not just availability with disclosure, but just making the data available regardless of whether the results are positive or negative when we are studying. And that goes for diagnostics or treatments. Whether or not it's published, that data should be available. And I think an important consequence of doing that and making this data available is to further our understanding of the biomarkers and thinking about the bigger picture of this Alzheimer's disease being on a continuum and understanding each phase and how we quantify the pathology and understanding. And biomarkers are going to play just a key role in this. So I think all of this sort of gets at this gaps, the gaps and opportunities, more or less of what we had drawn up before the session. And I think there's opportunities here to add to this slide as well afterwards. Anyway, I'll stop there. And I don't know if my other colleagues have anything else that they wanted to add to anything I missed.
Amber, Christina, is there anything you might like to add?
I think I was following Chris. It sounds you really covered it really well. One thing that the conversation ended was I found particularly interesting, so I wanted to say it. It's just this need for figuring out how to communicate to everybody, and maybe there's standard language or visuals that can be created to support the communication between like a researcher and the study participants, and that can help that giving of information back as part of the experience of being in the research.
Thanks, Amber. Yeah, and we, I know in the cognition session, thanks, Christina. In the cognition session, that was discussed as well, return of results. And I know one thing that's come up is, if you do have results, let's say you have a suite of biomarkers and they're giving you different information, and that different information is not necessarily consistent, or there's a lot of uncertainty, or their, measurement properties of different research tools are at different stages of development. How is information delivered in an understandable way that's true to the data that's being collected? And I think that will be something we'll continue to discuss at the general discussion. But I think Cynthia had some good ideas for what she'd like to see, being a research participant herself for a while. So I'm sure Cynthia can share some of what she shared with that group as well. All right, wonderful. And I'll turn it over to you now, Joe.
Thank you. So reporting, so we're not actually going to put our GNO slide up for our group because I have not been able to edit it yet. And we kind of went into a little bit of a different direction. I'm going to touch on rapidly 6 points, mostly coming from the breakout session. And some of these have already been said by the other groups. So number one, it's important to translate primary outcomes and combined outcomes from trials into terms that are both understandable and meaningful to patients. We learned, especially from the learned experiences, colleagues in the room, that what matters most to patients is to maintain your quality of life for a longer period of time. Well, time isn't the primary outcome and often not a secondary outcome of many of these trials. So how do we navigate that? There's debate on whether you can accurately, scientifically do a linear back projection on a nonlinear process. And yet that's what we're going to have to do. I would suggest maybe we're okay if we can say, look, to the best of our knowledge, you may experience a 5 to 7 month delay because you're this type of patient. We have less data. We're trying to accumulate that. That's why we're asking you to share some of this confidential data, anonymized, into these large real-world databases. And we'll collect more longitudinal data, and we'll get back to you on that. The second thing, ideal to have a core group of measures that we've all talked about that would also work for subgroups and for earlier disease. But that may be a tall order. And you may end up possibly with this common menu. There's your standardization, but it gets there's adaptive application to certain subgroups, as Dr. Gibbons was discussing the other day. Don't know. Third, digital health technologies may help in decreasing patient burden, but also providing more sensitive data, which is especially important for the very early disease. 4, connected to more data on subgroups, you need to have large real-world data and long-term assessments of benefits and harms. Number 5, to make use of that large real-world data, you need to have standardization of data and data sharing, and then more comparison between trials, more meta-analysis among different trials and studies and things like that. Finally, number 6, don't forget research to help patients with dementia, whether mild or moderate. There's the whole field is, appropriately focusing on earlier stages of disease. That may certainly be important if you're targeting amyloid beta, but don't forget to fund interventions that target other than amyloid to help some of these other diseases, related diseases, and then patients with moderate dementia. Thank you.
Thank you, Joe. Anybody else in the session have anything they'd like to add? Great. So a couple things that you brought up, Joe, I think we'll return to in the conversation, in general discussion. One of which is, as we move earlier, what are we talking about in terms of earlier, and how is that going to influence the measurement considerations we're all discussing? Does it differ for trial types? If we're thinking about pharmacological versus non-pharmacological trials, prevention, primary prevention versus secondary prevention, like how much do those considerations come into play when we're talking about some of the measurement considerations and challenges? Real world evidence, it'd be good for us to talk about what we mean a little bit more about that. We've talked about different ways of capturing different types of data in different contexts and different folks. So maybe getting a better sense of the types of real world data that we think are most impactful for what matters most to people, in what context, where we're missing data, that we need data, and how best to capture that with the measurement tools we have currently, but also what measurement needs there are to address gaps that we can't currently capture with the existing approaches that we have available to us. Great. So Sarah, I'll turn it over to you guys in the lived experience expert panel.
Thanks. I don't know if you still have that slide that oh, great. Thank you for bringing that up. I think what I'd just like to share that I took as a takeaway from listening to our experts in lived experience is that by asking communities and people with lived experience what's important to them, it's actually not only a method to build trust with that community, but as we give back to that community, it's a way to maintain that relationship over time. And I think what, as someone who sits in the clinical trial space, we have a lot of conversations about reducing participant burden. So studies like ADNI that are just annual or even sometimes every 2 years for people that don't have symptoms, we're thinking as we design these studies that we're minimizing burden on our participants, but what we heard from Rochelle and from Cynthia is that that results in a lack of connection. And a missed opportunity for relationship building, which is what they're looking for. And so we heard from Barbara that there are things that researchers can do to build those relationships, to maintain them over time, to design those interactions around the needs of the community and the desires of the community. So we know we can do it, but we need a real investment in that, not only, you know, funding, but time, which we know that all researchers are time poor, but it is critical that this be a priority for research. What one of our panelists asked me to share just on her behalf as we wrap is to remember that we are people with real feelings, not just a disease, and we need to know that someone will be there for us. And what we see with some of our academic research centers is a real division between the clinical and the research team, which now as we're entering into disease modification, that's felt by our research participants and by our patients. So I think more continuity and stronger connections between research and clinical will really go a long way towards ensuring that our participants are seen as more than just their disease. We see that clinical relevance is three-dimensional for our patients, for people with lived experience, for people that have elevated risk. So what's relevant to my brain impacts every aspect of our lives. And as we did some discussion in the everyday function area, that our current measurements of everyday function don't talk about what is so important, those social interactions, the ability to maintain meaning, communication. So there are some big gaps there, and I hope that we can really invest in those. And I think by somehow working closely with our participants, we have them as an incredible resource to tell us what is important and to make those modifications to our studies. I think I've covered everything here. I think, you know, I think what I'd like to do just in the final minutes is just turn to my co-chair for our Research Participant Advisory Board, Lupe Morales, who's been here the whole week. And I just ask Lupe if you have any thoughts that you'd like to add to that.
Sure. Thank you. It's been a pleasure to be involved. I still feel like I'm drinking from a fire hydrant sometimes, but I wanted to share something. And really, it's a rhetorical question to all of you. I was in the cognitive small group, and one of the things that was brought up is that these are opportunities for us to work across silos and develop working professional, multidisciplinary collaborations to share our work, to learn from each other, and to improve overall in a collective. And I'm wondering, and again, this is a rhetorical question because of the work that we do, how many of you are currently actively pursuing, involving research participants, whether at an advisory board or a community action board, but people with that experience or people who really want to be part of research? How and when are you involving them in the processes that you are involved in to improve research, number one? And number two, can they be part of that multidisciplinary collaborative team where we learn from each other and share resources with each other throughout the research methodology process? So I want to thank you. I'm not a scientist. I'm just hanging it all my life and what my experience is, but I do appreciate all of the comments and learnings from this workshop. Thank you.
Thank you, Lupe. And Barbara, Rochelle, Cynthia, do any of you want to make any final points? You're very welcome to speak up.
I just want to say thanks. This has been a very enriched 3 days for me to hear that I'm not alone, that somebody out there gets it. It's just that I wish they were a little closer to me. It would be easier. But, you know, as long as we have platforms like this where we can exchange from wherever we are in the country and the world, what works and what isn't working, that gives me hope for the future. And just wanted to say thank you. I appreciate it.
Thank you, Rochelle. Barbara?
I will reiterate that from Rochelle. Thank you so much. Let me have an echo here. I so appreciate this opportunity. Boston was my first clinical trials conference. Thank you, Sarah and Dr. Carrie Gleason for getting that together, and Tracy Smith for allowing me to be there. So I haven't had the history the other two participants have had. Rochelle and Cynthia have a longer history, but as someone who has been sort of an ambassador for Alzheimer's, prevention and education, I'm a dementia-friendly trainer, and I'm someone that people in the community know and connect me with because I talk about it all the time. And I will be talking about this conference. I talked about the previous conference and what I learned. And because of that, and I'm someone that is trusted, if I can work with their money, I have the trusting relationship, that's been cultivated over years. So this information is so valuable, and I will be sharing it. And I want you to know, I'll be sharing even the concerns and challenges you're having so that people have more empathy, and research participants have more empathy and understanding of what you are going through. Because sometimes we think of you as the ivory tower and that you're just doing the numbers and you're doing the research. Mm-hmm. And then, but now I have a whole nother perspective that I can share with them as well. And that you're working as hard as we're trying to work on doing our thing as being good participants. So I have much to share with them. I'm excited about continuing to be involved and offering back to you anything I can do to be helpful in this journey we're all on. Thank you.
Sarah?
Thank you, Barbara. Yes, please, Cynthia, go.
I would just say thank you to those who organized this wonderful workshop. And I just appreciate everyone coming on. And I really, again, urge you to look at our Research Participants Bill of Rights and embrace that as part of your research efforts. And thank you for listening to us. And we're here to help. One of the mottos that we often use as people living with dementia is nothing about us without us. So please do include us in your different boards and ask us. We'll tell you. Thanks for coming.
Thank you, Cynthia.
Thank you. Yeah, thank you guys once again for fantastic summaries and concluding comments. And Lupe, I will challenge you. We always talk about citizen scientists. You might not be the type of scientists that are portrayed in Hollywood on TV or whatever, but we're all citizen scientists. You're the expert of yourself, learning about yourself, learning what works for you all the time. And so I would challenge you to reconsider your identity as a citizen scientist. Okay, so we are now at the general discussion period, and we have a little over 30 minutes for that. And then we will wrap up and discuss next steps, and then we will adjourn. So now is a time for everyone, especially our panelists and moderators, but for everyone, including people that have not had the opportunity to express their voice or that may have shared something in chat that has not been raised in the discussion. Now's the time to do that. It's also the time, if you'd like to make comments on the report outs from each individual group, ask questions of those group leaders for clarification on some of the conversations that you weren't part of as being, a member of one group and not another. Now's the time for that as well. So I see on my screen, I'm going to serve as moderator here. I'm going to go by what I see. I see Dr. Cummings, Jeff first, and then Russ, I see you next. We'll start there.
Great. Thanks, Luke. Really great conference and great comments all the way through. One of the things I was thinking about today as I was listening was a charge that we have is to advance clinical trial methodologies in a way that would include some of this kind of thinking, research participant input, novel ways of measuring change. And that's actually very difficult to do, as you know. That's why the process is very conservative, because companies don't want to invest a lot of money in tools that are not very well vetted. But there is a pathway through the Clinical Path Institute to do changes and change outcomes. And I just would like to add that to the discussion, that something like the Clinical Path Institute might be a way to get this really great set of thoughts into a mechanism that could become new clinical trial outcomes.
Thank you, Russ, and then Michelle.
Thanks, Luke, and thanks to you and the NIA for putting this important conference together. Lived experience leaders who are leaders, not just experts, and thank them for coming as well. And so I put in the chat, and you have to scroll up just a little bit, a PDF of a paper that we did that addresses some of the stuff that kept coming up about we should survey patient preferences across the disease spectrum, and we've done that at Us Against Alzheimer's and the 80 PACE Consortium. We published a paper on that last year, and interestingly, it didn't show huge variations among the disease stages of the disease, from people who were preclinical and at risk all the way to caregivers of severe people. There wasn't a huge variation in those things. We did get a number of items. And it's ironic that I speak right after my friend, the legendary Jeff Cummings, because one of the things that did show up really clearly across the groups that isn't measured regularly in clinical trials, but truly important to patients, is neuropsychiatric symptoms. We tend to think of Alzheimer's as just being about forgetting stuff, but the stuff that often matters to people living with the disease, and then for people later in the disease with their care partners, is hallucinations, delusions, depression, feeling worthless, things that are not captured in memory tests, and they're not captured very well in function tests, but are captured pretty well in a test that Jeff developed called the NPI-Q, and there's one or two other tests out there that do that. So important to understand that Alzheimer's is not, and we're just talking about Alzheimer's, not the entire spectrum of dementias, but that Alzheimer's is not just a disease of forgetting. There's a lot more going on.
So thank you for those comments, Russ. And yes, I think we haven't discussed it enough. Neuropsychiatric symptoms, behavioral symptoms outside of what we think of as so-called cognitive function. And we have a lot of experts here that work in mental health, for example, and psychiatry, that probably could provide more comments. Michelle.
Yeah, thanks, Luke. And I want to echo something and maybe expand a little bit on something that Dr. Cummings mentioned about the Critical Path Institute. So I first and foremost think that in order for us to continue to expand our scientific knowledge, efforts in the pre-competitive space is really the way to go. We need to be able to pull all stakeholders in the room together to be able to have these scientific discussions. We need to be able to pull together and data share in that way, to really work together and bring and answer the questions that we've been discussing for the last 3 days. And so the FDA is very familiar with the work of the Critical Path Institute. I know that some of their COA members, their clinical outcome assessment team, has been listening for the last couple of days of this meeting, but they do have a new consortium specific to Alzheimer's. They are beginning to look at some task force under their rare neurodegenerative disease area for some of the more rare neurodegenerative Alzheimer's related dementia aspects. But they also do have a robust COA program and experience in the biomarker front. That is just one example. My bigger point is that the need of pre-competitive space work is something that will really help us the biggest in the long run by leveraging everyone's expertise and resources to continue to add to the science that is rapidly evolving every day. And I know that if we work together, we'll only achieve our goals in a more accelerated way by doing that.
Thanks, Michelle. All right, we're open to other questions of each other, of the group leaders for the sessions you didn't attend. we had identified some big themes around measurement. We can get into those if we'd like as well. But I'll open it up to the group first.
One thing that I think hasn't come up a lot that I would just like to insert into the discussion as well is thinking about not just return of results to patients, where we as scientists generate those results and give them back and say what we think they mean, but how do you enable patient-led innovations in science? So I'm taking that citizen science concept a step further, basically. So, you can think about a world where we're not just delivering our interpreted results to patients, but we're delivering them the data themselves. We're enabling patients to work with family members to identify their own patterns, right? Especially in a context of doing things like, high frequency sampling, where you're asking about everyday experiences. And now patient and patient communities can start to identify that, when I have this certain health condition, my cognition gets poorer, things like that, right? So things that are uniquely, I think patient communities are uniquely motivated and knowledgeable to do. And with digital tools, enabling access to the tools so that patients can design their own N of 1 type studies and giving guidance on how to do that. That can be something where you can now massively enhance the community of people who are doing these basic kind of investigations to inform the science overall. And I think this is something that is not actually that hard, given the tools that we have today and our ability to do data sharing. So I just want to put that out there.
Thanks, Laura. Sue, I see you next.
Thank you. I just wanted to ask a question about process, because I participated in group 2, and I was just wondering, when you put all these findings together, is there going to be any effort to sort of send it around to folks that participated to see if they had further feedback on a draft summary or anything like that? Because I would love that opportunity.
Yeah. So we everything's being recorded, which will have made 508 compliant. And once that's available, will be posted on the website. And then we will also have a science writers here from Rose Lee Associates, who will be putting together a workshop report based on everything we're discussing here. That will also be posted onto the meeting website, and it will be reviewable. We will continue, myself and Nicole Kidweiler, to serve as the main points of contact. Those are the only deliverables that the group decided on prior to the meeting. But now's the time. If I know some people have discussed comments about wanting to develop some of the G&O slides, for example, the gaps and opportunities slides, further to modify what was in there based on the discussion we're having now or over the course of the 3 days. So there may be opportunities to do that collectively. So if you have ideas for other next steps, at the end of this session, I will be giving a summary, but you can share the needs that you have identified here with the group. And this is a good time to do that.
Okay, well, I mean, my thinking was just when Dr. Hutter presented for number 2, we didn't re-show our slide. So it was hard to sort of see a visual of the different points. I was just hearing them. And I don't know if anybody else had that experience, but a lot of times it's easier when you can sort of see something to react to it. So to the extent that the points can be, sent around and folks can respond to them. But on ours, there were some points around point number 2 in particular. And then, it would just be great to have that opportunity. Thank you.
So the slides themselves, there's not a plan to post those. Those will help the science writer. That's really the purpose for them to be able to integrate those points into the final report. We can discuss with each group leader, because we didn't get permission for sharing those slides specifically in advance. We can make a note to talk to the groups to see if we can circulate those gaps and opportunity slides once they've been finalized. So I'm noted here for sure. I know other people have asked that question. Thank you. Siobhan, do you want to come on camera and mention what you put in chat?
Right, thanks. I can do, Luke. Yeah, I've just put a note in there to really remind us the importance of social health at all stages of the disease and way before it starts, and the opportunities really to measure that as well alongside the biomarkers and everything else. So, you know, I talked about it mainly in relation to what matters most to people living with dementia in the mild to moderate stage, but I think it's a lot earlier that we need to be looking at that, and in the preventing of dementia and staying connected. So I've just put a few references in there and a theoretical framework that that's been put forward and is really worth looking at.
Thank you very much. Very important points. And I should also say we are capturing the chat and trying to capture all the references that have been shared. So continue to please share those. Again, I encourage you to reach out to myself and Nicole after the meeting if other references or research that you feel is relevant to share with the group comes to mind. And then Russ, I see you have a follow-up for Dr. Riley. I'm reading it in real time here. Oh, okay.
Just noting that we did do some work on that, and it's showing up in our work as well. That's the domain we identified as social life / social activities. So it is showing up pretty clearly in our current research as well, Luke.
Thank you. All right. Oh, okay, I'm having, it's always a challenge to keep up with the chat when I get personal and group meets. So social health, one of the primary. Thank you, Rosie. I don't know if you want to expand on your comment, Rosie.
I just, there's been some research about the detrimental effects of social isolation, perceived loneliness, and these sorts of things. And so it's been looked at through those lenses. But when we think about all of us, or many of us, we go out into the community, people ask us, what do I need to do to stay healthy? And so we say things like, what's good for the heart is good for the brain. We say exercise is king, sleep, nutrition, etc. And we give all these pillars of health. Well, socialization, deep, meaningful social relationships. This is a pillar of health. We're human beings. We need each other. We're interdependent. And so social health, finding better ways to capture that is very important. And so I'm so glad. Thank you so much for the references. I'll be looking into that because it's, we're always trying to find ways to measure things better. And so I think moving forward, that's one of the things that have become very clear to me, at least, and highlighted by this conference, how important it is to better measure social health as a pillar of health.
And Liz Nielsen, do you want to follow up on your point or others?
Sure, I'm happy to. I think it's just maybe just a further reiteration of what's just been said and said so eloquently on the first day by Siobhan Riley, is that, the ability, we hear so much from folks in the lived experience panel about, and from the other qualitative research that's been done with them on the importance of being able to stay engaged and have function for a long period of time. But fundamentally what that means for most people is to be able to still be considered a meaningful social agent in the world. And so, assessing people's everyday function goes well beyond assessing their sort of mental health capacity or ability to, perform IDLs and ADLs, but actually to engage meaningfully in interpersonal activities and in family and social relationships. And so I think that any assessment of everyday function that doesn't take that seriously would be impoverished if we want to gather what matters most to the people living with the disease. So I appreciate the talks and comments around that theme. Thank you.
Thanks, Liz. Yeah, Suzanne?
I would be really interested in knowing whether there are databases that have collected this kind of information over time, and whether we could have those potentially made available if we were to start a group, maybe work through CPAC, wherever the best place is. And one of the things that that is relevant to that type of an effort is that in addition to having time savings potentially be something that can be used to explain the benefit of treatments to people and show what kind of an impact you're having, it's also a common metric where you can translate any of these types of outcomes into a time savings or how much time passes as you lose these kinds of functions, which makes it an easy way to bridge from one database to another, even when you don't necessarily have common metrics across them, because the time in disease is a common metric. There's some things you have to do to align the populations and things like that. But what that allows you to do is talk about which aspects of all of these important domains that are important to those experiencing Alzheimer's disease, which aspects are really progressive, and then which ones are maybe not as progressive, but might still be subject to treatment effects. And so again, it's sort of separating this disease modification from symptomatic types of effects and symptomatic or daily concerns that maybe aren't necessarily as progressive. And with the progressive outcomes, specifically the time metric can help bridge across different databases. And if we had databases that were measuring all of these different types of things, we could put together what we think could be, a really good scale that includes outcomes across many different types of scales by aligning them on that time axis.
Thanks, Suzanne. Ron.
Alrighty, thanks, Luke. Just going back to sort of the theme of the meeting, clinically meaningful outcomes in AD and ADRD trials. How do we measure these things? So sort of what Suzanne was saying, I'm wondering if we can come up with mechanisms for actually developing and validating these instruments that may capture what we've been discussing over the past 3 days. And Jeff was alluding to the fact that the companies are very, very reluctant to put these measures into trials if they haven't been validated. They're putting a lot of money in the trial, and I'm not sure I want to use this particular instrument. So I think we need to think about mechanisms for doing this. CPATH may be one. Maybe your division at NIA could be developing certain instruments, or maybe you do have RFAs out there for development of instruments to capture these constructs that might be usable in clinical trials, so that we actually are able to validate whether or not our interventions are having any meaningful effect on these important constructs.
Thank you, Ron. I'm going to share something at the end of the meeting on one particular opportunity. We have several. I would encourage people to check out our funding opportunity page. I don't know, Nicole, if you're on, or Elena or someone, if you want to share our division funding resource page. This is a new opportunity I'll talk about towards the end of the meeting, Ron, to put together a measurement coordinating network, which also includes measurement validation. I see Andrea and Suzanne's hands up, but before we go to you, to address your comment, is something that we talked about briefly in the report outs, but validation, what that means for what context of use. I don't know if others want to comment on that particular point. So if someone's looking at a set of measures and they're considering validation, what does that actually mean? So that came up in conversation. We didn't dig into the details there, but I think that this term's often thrown around, kind of like I was bringing up real world data because it's another one to me that it means a lot of different things to a lot of different people. So understanding better what people mean by that, what the gaps are, I'd be interested to hear from others about that now, or before we close. That would be good to hear.
So that was one of my comments that I was going to talk about, which is, that there are some good publications out there that show the different aspects of validation that are important, and it's almost a list of all the different types of validation that you could do. And about 5 years ago, I was on a committee that had some NIH members, FDA members, and some industry members, and we worked on specifically statistical approach to validation of biomarkers and what all the different steps of that biomarker validation is. So I'll share that link in the chat. And then the other piece of it was just to say, there is kind of a standard list of things that you need to do to be able to show the scale is validated. Interestingly, it's very hard to have a sensitive progressive measure that isn't already reliable and isn't already valid in some aspects of validity. There's other aspects you still have to specifically address, but if it's already a good measure of progression with small variability around that measure, then there's certain properties that it comes with. And certain aspects of validation that then you don't have to necessarily directly assess, but could indirectly assess through that measurement of progression. But I'll post that link for the biomarker validation. We worked on it for probably 4 years. It was a very long and tedious process. And it's a very detailed document, and it goes through all the different steps of biomarker validation, starting from actually validating the assays or the metrics from the beginning all the way through how it can be validated for specific contexts of use.
Thanks, Suzanne. Yes, please do share that. And I see Jeff and then Cheryl.
Yeah, so Luke, I think this idea of context of use is so important. There are 7 recognized categories for context of use by the FDA in the BEST catalog. I think pre-planning that when a tool is being developed. So is this a diagnostic tool? Is this a monitoring tool? Is this a safety tool? Really affects the characteristics of the test that is being developed. So I think as we think about how to institute some of these ideas into trials, then the context of use in the tool, in the trial, where it becomes a drug development tool is really important. That's one of the shaping concepts that needs to be identified before the tool is developed or applied.
Thanks, Jeff. Cheryl?
That was a beautiful summary by Dr. Cummins. Thank you for sharing that. The perspective I wanted to bring here, I do work for Critical Path Institute. I've heard our name come up a few times in the last 20 minutes or so. So I did want to speak up on the Vice President of the Clinical Outcome Assessment Program. And we are working both on the COA tools as well as biomarkers at CPATH with a regulatory lens, thinking about the context of use in clinical trial endpoints. And what we find when we're thinking about validating tools is the rate limiting factor is data. And so when we're identifying things that we want to learn more information about, the most important thing we can do is to get that tool into studies, to start thinking about data collection on day one, because the sooner we can get data, the sooner we can have the evidence that we need, and the sooner we can make the decision based on that evidence. So for any of those of you out there who are running your own studies or thinking about an upcoming study, I would ask you to connect with the folks, the thought leaders in the field on either the COA side or the biomarker side, and work with them to identify things that maybe you haven't thought of for including in your own trial and your own studies, so that we can come together as a field, collect that data, and be able to find the answers that we need with data-driven approaches.
Thanks, Cheryl. Let's see. And I saw in chat, thanks, Suzanne, but folks from NINDS and NIA, the Division of Behavioral and Social Research, have shared some links to their funding opportunities as well. So I'd encourage everybody to check those out. Joe, I see your hand.
Yeah,
You're on mute.
There you go. I imagine that rate limiting factor would apply to, you're looking for randomized as well as non-randomized data in there. Is that correct? For instance, we're not going to be able to, like the overall trial results may demonstrate X on the primary outcome and the secondary outcomes. And then you look at the forest plots of subgroups, and it's pretty scary. And sometimes forest plots get over-interpreted, right? And when you're filling in those, it may not be possible, some have said, don't disagree, to then do a prospective randomized controlled trial on every single subgroup possible under there. So can you capture and get the data you need on the quote unquote real world data or registry based data platforms and things like that, EHRs, real clinical use? Is that going to be helpful? Even though that's not randomized, you do have a foundation of randomized data, but not everything in the future is going to be randomized.
I think that was directed to me, so I'm going to jump in. Interventional trial data is probably the most powerful way, because you know if it's an effective therapy, there's going to be change over time. But non-randomized, non-interventional observational data can absolutely play a role in understanding how these tools function. It's the longitudinal nature that we're really looking for. So if it's a natural history study, we know with this type of population, there will be changes over time. So as long as there's change over time for us to be able to anchor to and then see the changes in biomarkers or see the changes in COA scores, then we should have the type of evidence that would be able to give us an idea of if the tool is actually going to be useful then in a clinical trial setting or in other settings.
Thank you. And maybe, I don't know if Robert Gibbons is still on, but we had also discussed, challenges. You can't, if you're doing measurement development and validation, you can't do that in every single, potential subgroup of importance in every single context. And there are ways of using newer adaptive measurement techniques, for example. Robert, I see you now. Do you want to make a comment based on that?
Sure. Both the question of randomization and validation. Typically, I think the point is that you don't need randomized controlled trials to validate a new measure, but it is important as one aspect of the validation to make sure that the measures are sensitive to change. And there you absolutely need longitudinal data. So echoing what Cheryl said. The idea of differential item functioning, which is not really a part of adaptive testing, it comes out of item response theory and multidimensional item response theory, which is really important as a distinction here because you're dealing with complex traits, is that we don't have to validate in every single subpopulation. We have good statistical approaches if we have good calibration data in a general population to determine whether or not the item parameters are invariant with respect to the general population, or if there are subsets of the items that just function in a very different way. They're not good discriminators of high and low levels of the underlying ability metric. And for that, we don't have to go out and do lengthy validation studies. We can learn that kind of information from estimating the item parameters in that subpopulation and comparing them to the parent population. So we can very quickly determine whether or not there's a subset of the items that are biased. And one should focus on that as a very practical way of tailoring the quality of our measurements to specific populations, in particular minoritized populations.
Yeah, I guess I was using the adaptive example. I was thinking about the conversation with Cynthia about meeting people where they are and thinking about what's meaningful to an individual. How do we, think about measurement, validation, and testing in the context of getting us closer to a personalized approach to assessment and intervention? But your points are well taken. So any other comments about that? Or Andrea, I see you have your hand, maybe
it's a different
point.
It's a different comment.
Yeah, that's what I figured. Go ahead.
Are you sure? Does anyone have anything else on this? I don't want to.
I can just respond to your point, and then I'll do it very quickly. Yes, absolutely. That you're back in the realm of adaptive testing, and the ability to bring items that are geared to a particular person's level of severity is critical for measurement, because you just burn out if I'm asking a fourth grader lots of calculus and real analysis questions. They're math questions, but they're not well suited to that person. And everything done in this field is exactly that. You're running the gamut, giving everybody the same set of questions, irrespective of where they are in terms of their cognitive ability or potential impairment. Sorry,
Thanks.
now I'll pass it over.
Thanks. Andrea.
I'm enjoying this conversation so much. I had a new thought that I wanted to introduce as we're closing. And I've really been reflecting. I've so enjoyed this workshop just in sort of the past, I don't know, almost 2 decades of things that I've seen in different discussions. And certainly not everything about the lack of meaningful outcomes is attributable to this, but some of it is that we have not done a good job including people living with dementia in the research process. And I think that if we had more consistency with which we did that, you know, I feel that we are always learning and discovering, and it's so insightful. And I think it's important to state that the US is very behind in involvement of the public in research in terms of standards that we have and that we place on ourselves. And, you know, this is a wonderful meeting where we're seeing how feasible that is yet again. And I think the NIA has been doing a great job in other meetings and in summits. And I know the Alzheimer's Association and others are doing a great job with this. So I think I just don't want us to lose that component in all of these things we're so excited to do. And just that we do have the opportunity to integrate as we work on these new measures in different cycles, that perspective. It doesn't have to just come at certain points in time when it feels convenient. But I think what we're learning and what we can see is it's helping us think about better scientific questions. So I just want to make sure that we keep track of that as we go forward and get really excited about the things we're going to do, because I think we've heard a lot of people tell us that they would like to be included, you know, not sporadically and not as a convenience. And I think we have lots of evidence that this is highly feasible. And I think people weren't convinced of that 20 years ago, but it's very, very clear. So I just wanted to make sure that we remembered that piece.
Thanks, Andrea. Do any of our leaders from any of our sessions want to make any concluding remarks? We're at time for wrap up and next steps. Nothing about us without us. Thank you, Cynthia. Okay, so I will wrap up. I already mentioned what you can expect at the conclusion of this meeting. These recordings will be made 508 compliant, and we will have a workshop report that will be posted to the website. Behind the scenes, there are also some other opportunities, which I won't bring up now, but opportunities for maybe writing a paper in addition to just the workshop report with a group of folks that might be interested in that. And then we also might have a debrief to develop those gaps and opportunities slides to finalize them and chat with the group about potentially sharing those as well. I think people found those to be very valuable. So any other ideas folks have for next steps, you don't have to voice those now, but again, please contact myself or Nicole Kidweiler or both of us, and we can bring that information to the planning group. Jennifer, can you go back to the first slide? Yeah, that one. So I want to thank all of you. This has been a really, really fantastic meeting, and it's, you know, entirely due to the participation that has been so broad and the diverse set of voices that have continued to bring us back to what is meaningful in the context of the trials that we're all interested in conducting to find cures for the diseases that we're all here to discuss. I want to again thank especially the planning committee that over the last almost year has put in work to think about the issues that all you expert folks have come to discuss today. Again, Christy Hardy, and this is a team at NIA and NINDS, including Christy Hardy, Melissa Trevino in the cognitive function session, Elena Fazio and Benford Williams in the everyday function session, Amber McCartney and Christina McClendon in the biomarkers session, and Priscilla Novak, Marcel Salive, and Nada Radoja in the payers, regulators, and health economics session. Can you go to the next slide? We have shared some opportunities for new research, and this is one among many. It's a new opportunity. We'll have a webinar coming up March 29th, so later this month. And I already shared a link in chat, but this is essentially an open measurement coordinating network. This is focused on primary prevention trials, but a lot of measurement, development, and validation from mechanisms that account for why someone does respond or not to a given intervention in a given context, as well as outcome measures. How do we know that changes are happening that are meaningful? So I think all of these considerations expressed here will be an important part of that project. And to address the points that were raised, this is very much focused on the input of the research participants as well. So built into this is a community advisories panels and other expert input to help shape this network. It's very much a cooperative agreement, which means that NIH will work with the outside research team, including community members, to make this successful. Can you go to the next slide? This is a couple activities that we've listed as potential areas of focus. It's not exhaustive, but it gives you some ideas about the types of things we hope to accomplish. You can close that, Jennifer, that's fine. And so unless there are other comments, I see some very nice comments in the chat. I'll let everybody read those and absorb them, but we are at close, and that's pretty much it. So thank you again, and have a great day.
Thank you, Luke.
Yep, you're welcome.
Thank you.